ते बंद करा.
Good morning, all of you. We'll start within 5 minutes that all the participants could easily join us.
आवाज कोणता तुला आहे
आले पहिले आले पहिले आले पहिले
I once again welcome you all to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates from all over. I once again 2 minutes to go to all the distinguished delegates. I A very warm welcome and good morning to the galaxy of intellectuals, dignitaries, invited guests, distinguished delegates from all over, and my colleagues. I, Dr. Rupali Tekade, Assistant Professor, Department of Zoology, feels privileged to extend my warm welcome on behalf of Shri Datta Prasadik Shikshan Prasarak Mandal, late Rajkamalji Bharti, Arts Commerce, and Srimati Sushilabai Rajkamalji Bharti, Science College, Arnis, IQSSL, and Department of Botany and Zoology in one-day national e-seminar on COVID-19 impact on life sciences. I feel honored to announce that for this webinar, we have Dr. Ravikiran Govekar, Chief Conservator of Forest and Field Director, Pench Tiger Reserve, Nagpur. We also have Dr. Mumtaz Beg, Department of Integrative Biology, University of Guelph, Ontario, Canada. Dr. S.R. Yadav, INSA, Senior Scientist, Shivaji University, Kolhapur. We also have Dr. Ashish Kumar Jha, Assistant Professor, Department of Zoology, Hislop College, Nagpur. Last but not least, we have Dr. DVNS Suresh, Professor, Department of Zoology, Dr. Ambedkar College, Nagpur. I again welcome all these eminent speakers, guests, delegates from all over the country who have joined us from the different walks of their life on behalf of organizing committee, organizer, and the organizing secretary. Every year, our college organizes conference, workshop, and seminar. But this year, this seminar holds a special value because, as we all know, that due to the COVID-19 pandemic conditions, we are organized this on a very digital platform. The theme for this webinar is SARS-CoV-19 scenario in India, a genomic insight, plant diversity of India with special reference to antiviral species, immune system, and coronavirus, COVID, understanding and combating in a better way. Moving forward very quickly, COVID-19 is a pandemic prevailing across all over the world past few months. Every day it seems to reach a new and a tragic milestone. Every day we are learning more and more about the virus and the diseases it causes. So for this, we have our expert speaker from across the field who will be sharing their knowledge, thoughts, research works, and opinion with this. But before this, I will simply give some housekeeping items. I request all the attendees to please mute your cell phones. If you have any questions regarding the presentation, you can simply type it into the chat box or your Zoom control panel. And the last thing is that the filling up of the feedback form is very necessary because it is very important to you as well as us also. To start with, I feel very happy to call Professor Milind Pandey, IQSC coordinator, for his introductory speech. Over to you, Pandey sir.
Thank you very much, madam. Good morning, friends. Myself, Professor M.M. Pandey, IQSC coordinator of late Rajkamalji Bharti Arts, Commerce and Science, Arts, Commerce, and Srimati Sushilabai Bharti Science College, Arne. First of all, I welcome all of you, all the guest lecturers, subject experts, all the participants all over the world. President of our college, Dr. Sanjay Bharti, Secretary of our institution, Siddharth Bharti, Dr. R.M. Tated, the principal of our college, and all my teachers and non-teaching staff in the national webinar on COVID-19 impact on life sciences, organized by Department of Zoology and Botany, teachers collaboration with IQSC of the college. National webinar. Friends, COVID-19. All of you know that due to COVID-19, this world has been divided into two categories, two ages. The world before COVID-19 and the world after COVID-19. All of you know that science COVID-19 deeply concerned human life. Friends, I am happy that to show that the college and institution always takes initiative in various valuable social activities deeply concerned human life. We had organized various awareness programs regarding COVID-19 in March 2020. Under the leadership of our President, Dr. Sanjay Ji Bharti, who is the head of the Department of Botanic Department at Government Medical College, Yavatmal, but also under the leadership of the college, we provided the kits to the police department, doctors, nurses, and also provided masks to the saloons and grocery shops, and also contributed in government relief fund. Almost all our employee contributed in the government relief fund. Thus, we actively extended our contribution to the social welfare of the country. Today's webinar is the step ahead towards the social contribution. Being the IQSC coordinator, I wish you all the best for this webinar and thank you very much. I hand over this mic to Professor Tekade Madam. Thank you very much.
Thank you, sir, for your introductory speech. May I now request Dr. R.M. Tated, our beloved principal, late Rajkamalji Bharti, Arts Commerce, and Srimati Sushila Bai Rajkamalji Bharti, Science College, Arni, for his welcome remarks. Over to you, Dr. Tated. Science College, Arni, for his welcome remarks. Over to you, Dr. Tated. Science College, Arni, for his welcome remarks.
Good morning, friends. Myself, Dr. Rajendra Tated, the principal of late Rajkamalji Bharti Arts Commerce and Srimati Sushila R. Bharti Science College, Arni. Welcome you all in the national webinar on COVID-19 impact on life science organized by the Department of Botany, Zoology and IQS. Friends, our college was established in 1984 with two faculties, arts and commerce, and then we started science faculty in 2009 under the leadership of our president, Dr. Sanjay Ji Bharti, head of the department orthopedic department at Government Medical College, Yavatmal. The main purpose of starting science faculty was to give the girl students in our area on opportunity of studying graduation in all the faculties. We also faced NAC in 2016 successfully. Our college takes initiative in many social activity like blood donation camps, medical health checkup camps, women empowerment programs, and other many awareness programs on social problems. Our college also organize many workshops, seminars, and conferences in the college, and also planning to organize many conferences of various subjects in the future. Today also we subject in the future. Today also we subject in the future. Today also we have organized the national webinar on COVID-19 impact on life science. Being the principal of the college, I feel very glad to let you know that though our college is in the rural area, almost 600 participants from all over the world joined this webinar. So I am very grateful to you all. I again welcome you all in this webinar and wish you all the best and good day ahead. Thank you very much. Thank you very much. And wish you all the best and good day ahead. Thank you very much. Thank you very much.
A very big thank you to you, sir. Your each and every word gives us energy to work more and more in a better way for the institution. Now, friend, it's time to request Dr. Sanjay Ji Bharti, President, Sri Datta Prasadik Shikshan Prasarak Mandal, Army, a man of distinct vision and a fountainhead of illuminating ideas, an idol of knowledge and experience. I request Dr. Sanjay Ji Bharti to please have his presidential address for today's seminar. Over to you, Dr. Bharti. I request Dr. Sanjay Ji Bharti to please have his presidential address for today's seminar.
Arranged by Sri
Over to you,
Datta
Dr.
Prasadik
Bharti.
Shikshan Prasaran
I
Mandal,
request
Dead
Dr.
Morning,
Sanjay Ji Bharti
Rajkamal,
to
Bharti,
please have
Amir
his
Commerce
presidential
College,
address
Shilabai,
for today's seminar.
Science College. It is arranged by immense pleasure
Over to you,
that Datta
Dr.
Prasadik
Bharti.
Shikshan Prasaran
I request
current
Dr.
and
Sanjay Ji
emerging
Bharti
topic
to please have
COVID-19.
his presidential address
Much is
for
talk
today's
regarding
seminar.
this COVID-19 arranged by immense regarding the electronic media and printing
I
media.
request
Current
Dr.
and
Sanjay Ji Bharti
emerging topic just COVID-19. Much is talk regarding this COVID-19 arranged by topic COVID-19 electronic media. You know that banking has faced this pandemic after much is talk regarding COVID-19 arranged by electronic media. You know that banking has faced this pandemic
Sorry for the inconvenience. Due to disturbance and lots of echo, we are moving forward. We are again trying to be with Dr. Sanjay Ji Bharti. We are moving forward. I once again request sir to please join us and have his presidential address. We are again trying
Good morning.
to
Today,
be with
very
Dr.
glad
Sanjay
that we
Ji
are
Bharti.
taking a
We are
webinar on
moving forward. I once again
COVID-19
request sir
arranged
to
by
please join us
Sri Datta
and have his
Prasadik
presidential
Shikshan
address. We are
Prasaran Mandal,
again
Dead Morning, Rajkamal, Bharti,
with
Arts and
Dr.
Commerce
Sanjay
College,
Ji Bharti.
Shilabai,
We are
on
moving
Science
forward. I
College.
once again
It is
request sir
arranged
to
by
please join
immense
us
pleasure that Datta current and Rajkamal, Bharti, Arts and Commerce College, Immense pleasure that the electronic media and printing media current and opportunity of just the astronomy to throw or to enlighten regarding this pleasure the electronic media. You know that mankind has faced opportunities of just regarding this plague, the electronic flu, and now that mankind has faced opportunities of just regarding this plague, now that pandemic, we have never expected that the planes and all planes are traveling stop for a period for 2 months, and we are not assessing. We have never lost. Just we have now assessed traveling stop for the human life. We are assessing human life. Assessing being a doctor, just we have now assessed. I have personally and one time for a COVID was life in the we can develop our immunity by very few things like yoga, pranayama, and these are just like no lemon, how lemon can develop our immunity by very few things like yoga, pranayama, and all and it goes on repeatedly going on mutation. That's why all these scientists and all these ICMR and various scientists yet to find a concrete solution. Deadly goes on mutation. There is no such medication yet found. Therefore, my request wearing the mask in that European and Western countries open there was no restriction in the second wave has declared that first European and Western all the drugs are resistant open there was no consumption in the second wave has the developers are European all the drugs to have this just
Thank you, sir, for your enthusiastic words. Now let us have the pleasure of listening to the inaugural remark. Starting with the session, I would like to request Professor Muneshwar, Assistant Professor, Department of Botany, to introduce Honorable Dr. Ravikiran Govekar, sir. Over to you, Professor Muneshwar.
this should be more careful than the older generation, and the children are more affected. Either in
A very warm good morning to everyone. Myself, Ashay Shamrao Muneshwar, Assistant Professor of Botany in Rajkamalji Bharati Art Commerce and Srimati Sushila Rajkamalji Bharati Science College, Army. I feel myself very lucky and privileged to introduce such a great personality, Dr. Ravikiran Govekar, sir, Indian Forest Services, Maharashtra, 2001. He was a gold medalist in BSc. He has completed MSc Botany from Shivaji University, Kolhapur, PG Diploma of Forestry and Wildlife Management from Dehradun.
He has completed his PhD in botany from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad in 2016. He has qualified various state level examinations and national level examinations like NET, SET, GATE, MCET, and UPSC. He has got professional training from Lal Bahadur Shastri National Academy of Administration, Mussoorie in 2001. He also obtained his professional training from Yashwantrao Chauhan Academy of Developmental Administration in Pune in 2001. Along with, Sir has got other important professional training from IIM Ahmedabad in 2009, Colorado State University in USA in 2010, Bio Academy in Finland, and government organizations in Malaysia in 2009. He has participated in various media interactions with All India Radio, Doordarshan, DD Sahyadri, G News, and Vidarbha News. He has written columns in well-known magazines and newspapers like Daily Lok Satta, Divya Marathi, Times of India, Green Hope Magazine, and The Hitwada. He has published two books which are named as Field Guide to Plants of Sahyadri and A Field Guide to Plants of Vidarbha. He has published more than 11 research papers on floristic and ethnobotanical studies with the description of 4 new plant species. 2 of them are named after the district Gadchiroli and Gondwan. Sir has worked as lecturer in various colleges such as DY College Patil Pune, Shivaji University Kolhapur, Bhavans College Mumbai, and Pre-IAS Training Institute, Government of Maharashtra. Sir has worked as Deputy Conservator of Forest in Gadchiroli, Assistant Inspector General, National Tiger Conservation Authority in Nagpur and Guwahati. He is on deputation to the Ministry of Environment, Forest and Climate Change, Government of India, with the administrative jurisdiction of the states of Maharashtra, Madhya Pradesh, Chhattisgarh, Odisha, Bihar, Jharkhand, West Bengal, Arunachal Pradesh, Mizoram, and Assam. He is conservator of forest and field director, Navegaon Nagzira Tiger Reserve, Gondia. Currently, he is working as chief conservator of forest and field director at Pench Tiger Reserve, Nagpur, and he is member secretary, Maharashtra State Zoology Authority. Now, without taking much time, I would like to request Dr. Ravi Kiran Govekar sir to deliver his thoughts. Thank you. Over to you, Dr. Govekar.
Yeah, good morning and thank you very much. Am I audible? Am I audible?
Yes, sir.
Yes, sir. Yes, sir.
And I'm visible too?
Yes, sir.
Okay, I request all the participants and maybe some other this one to mute their mics because there are lots of noise, background noise, and unfortunately we couldn't hear properly Dr. Sanjay Bharti's speech. So request everyone to mute their mic, please. Yeah. Thank you very much for my introduction, very long introduction. Especially, I thank you, Dr. Vinod Varghese. He has been in touch with me since last or 15 days or so. And of course, Dr. Tated, the principal of late Rajkamalji Bharati Arts Commerce and SR Bharati Science College, RNA. So it is really great pleasure for me to be here for 2-3 reasons. First thing is that this is my third or I think fourth inaugural speech for topic related to this, because in so many places this is going on because people are getting sensitive about this issue. They are getting more and more aware about this issue, and many colleges, education institutes are trying to sensitize their students, other people, and contribute in the social cause of getting over from this COVID epidemic. So the first reason was that RNE is the place which is again related to my administration. Since last 3 years, I have been associated with Tipeshwar Sanctuary and Penganga Sanctuary in Yavatmal, and I used to manage those areas till March last. And we had wonderful tiger experiments. You might be knowing that the tiger that has traveled longest distance of 2,000 kilometers from Yavatmal to Nanded to Adilabad and Pusad and Akola, all the way to Aurangabad, Ajanta, Ellora, and they came back and now settled in Dhanaganga. That global record was set up by the tiger from your area, Yavatmal area. Then there is another sanctuary, Painganga, which is close to you. Then there is third sanctuary, Isapur, near Pusad, that is also near to Arani. And if you travel south, north, west, then there is another sanctuary, Katepurna. So Arani is very strategically located, surrounded by these important sanctuaries. So being a forester and being a witness of that area, our tiger has also traveled from Pusad, Arani area towards Marathwada. So I thought it's fine for me to be there because I also undertake administration for that area. Secondly, during my introduction, might have heard that I had been a teacher before joining Indian Forest Service. I taught lots of students in different universities and colleges. Although it is for a very short period, but I taught, and whenever there is any program of this kind, I always feel privileged that I go back to my earlier profession and I get connected to the students and the teacher. And lastly, of course, the most important thing is that if somebody is doing something for COVID-19, for sensitization, awareness, or whatever measures or whatever activities, then everybody must contribute to that. So with all these three purposes, I am here. So I once again thanks the organizers, then Dr. Tated and Dr. of course Sanjay Bharti, the president of the Datta Prasadik Shikshan Prasar Mandal Arni, and all the faculty members over there. So today also you got the wonderful faculty members or these presenters here who will be telling lots about the plant world of the plants, then herbal medicines, and of course COVID and other things. Dr. Mumtaz Beg, Dr. Suresh, Dr. Jha, and of course my teacher, Dr. S.R. Yadav. He taught me a lot about taxonomy and he is still teaching me. So he's there. So I'm very happy that you will be getting benefited with all his expert advice, comments, and knowledge. So COVID-19, as you know, every nook and corner, everywhere in small village, town, gully, or country, or even President's Palace, everywhere there is only one discussion about this particular thing since last 6-7 months is COVID-19 or Corona. So everybody is trying to fight this epidemic. Everybody is trying to find some solution, and various measures are being taken up, and certain activities, they have been modified, certain things have been changed, certain approaches they have changed, and certain areas which are come in the focus, they got the priority, they got kind of limelight. And one of the focus areas is the life sciences or biological sciences, because everybody knows it's Corona, it is COVID-19, the SARS kind of virus, and we can't control it as long as we don't have a vaccine against it. And of course, there are lots of mortality. More than 6 crore people have so far infected globally. More than 1.4 million or 14 lakh people, they have lost their lives, and it's still going on. So as long as we don't have vaccine, effective vaccine against that, nothing can be done much, except that we can do some preventive measures, and we can just try to stop its spread further. If you are aware that today also the Honorable Prime Minister of India is visiting Pune and Hyderabad. Pune is visiting the Serum India Institute, then Bharat Pharmaceuticals, and of course Zydus Biotech in Ahmedabad. So when the Prime Minister of India visits some institutes which is trying to develop the vaccines, then and one day he's visiting 3 different laboratories. That itself indicates how important this particular subject is. It itself indicates how seriously the policymakers and administrators are taking this particular menace or particular epidemic nowadays. So this is how things are going on. In India, since we follow different streams of medicine, along with allopathy, we follow the Ayurveda also, we follow the Siddha medicine also. And Ayush Ministry, our government of India's Ayush Ministry, he tries to integrate the different types of systems like Ayurveda system or Siddha system, Unani system, along with, of course, allopathy. and lots has been have been discussed about, and lots of publications, lots of measures have been taken by Ayush Ministry. Guidelines are issued by the Ayush Ministry, and even Unani system also has certain medicines for this COVID and all. So biological systems which are based on the plants like Ayurveda or Unani or Siddha, they have to play a major role basically in preventive medicine, basically to stop the further spread of this disease. And of course, we have to take antibiotics, you have to take other medicines which are prescribed by the allopathic system, and everything goes well, then almost 60, 70%, 80% patients are coming back, returning. But thing is that in future, we have to depend more on our own health, how to improve our immunity, and of course, how to follow the newer approaches or newer avenues as modified after the COVID-19 epidemic. So reimaging the life sciences means we have to talk about our, you have to sensitize people about the disease and pandemic. You have to talk about the immunity. You have to take care of your comorbidity and age-related problems. There are newer normals. Now everything is now online. That is new normal. Lockdowns are very common. Home quarantine, webinars, virtual meetings, e-class, e-pass, e-pass. So all these things, they are now become buzzwords nowadays. So these are the new normals in our life. Then we talk about application part of the science. We normally do a lot about theoretical part, but we now talk about the application part of the science. Then along with allopathy, now we are giving slight importance to Ayurveda, research in Ayurveda. Then earlier you are supposed to go and get a medicine and get advice, but now sometimes you have to call your doctor and get a kind of telemedicine, some teleprescription and kind of thing. Then they talk about the nutraceuticals, they talk about the molecular medicine or this nanomedicines kind of thing. Then again, for students or for certain people, we talk about the psychotherapy, we talk about the stress related, because earlier days, I can share one funny incident. There is one small kid who is our neighbor. So when there was a lockdown, complete lockdown and social distancing, then she used to insist coming to our home. So we used to tell her that, okay, don't come because there is kind of corona monkey moving around. So corona monkey was a new word for children to tell them that, oh, it is something serious going on around and you should not come out. So that way, some new terminologies, new things are coming up. Then at policy level, also so many things are coming up. Just now during discussion, principal sir told that they have distributed some masks, some medicines, some other sanitizers kind of thing. Same thing happened everywhere in forest department also. Normally we safeguard our forest, but this time we provided lots of masks and sanitizers and soaps to thousands of people, which is not our job. Of course, providing masks or providing medicine is not a job of any college, but they are doing it. So there is a change in approach, there is change in policy. Everybody is seeing it as a work of social activity, and everybody has to participate in that. There are lots of changes in approach of the institutes and individual, and of course government. And lastly, if you can see, there is a lot of change in approach towards the health sector. Outlook towards doctors and nurses, health sector, investment in health sector, facilitation health sector, new facilities and centers are coming up, and there is change in the approach of research as well. So many biopharmaceutical institutes and organizations like NIV and Bharat Biotech, or there are so many Indian and foreign also, Russia and US and so many countries and their organization, they are very, very actively working on this particular thing. So this way, we are trying to deal with the corona, and of course the colleges, their biology wing, the botany, zoology, microbiology, or life sciences, whatever subject they are teaching, they are in focus for different purposes. One of the things, for example, if it's botany department, then the botany department and their scientists, their teachers, their professors, they can always explore if there is any local knowledge nearby, use of certain plant for maybe anti-malarial, anti-fever, or anti-cough kind of agent if it's there. If certain village or certain area consuming certain drugs or certain medicines, if it is not prone to the disease like COVID kind of thing? What are the active ingredient of certain plants which are being used by the tribal people, like Kolam people stay in, there are lots of Kolam people in Yavatmal. So what medicines are being used by the Kolam people in Yavatmal? So that way, science departments, botany departments, or zoology departments, or microbiology department, they can explore certain things and contribute in this nation building activity. And on that background, this particular workshop will definitely open up new thought process among the participants, the students, the researchers, and general public. So I think Dr. S.R. Yadav sir will be speaking next. That is, as usual, it will be a wonderful lecture. Last time also I enjoyed listening to his lecture, and all other speakers will be telling you more about this COVID and newer approaches. So with this, I will stop because lots of discussion is yet to come. And I again thank Dr. Varghese, Principal Dated, and Dr. Sanjay Bharati, and all the organizers for inviting me here and allowing me to express my thoughts, whichever I felt proper. So without losing much time, now I will announce this webinar as open and hand over the session to the organizers. Thank you very much.
So friends, this was Dr. Ravikiran Govekar, Chief Conservator of Forest and Field Director, French Tiger Reserve, Nagpur. Sir, I am very grateful to you for your valuable insight in the area of medicine or medicinal plants and its application. Thank you, sir, for sharing your knowledge and opinion. Thank you, sir. If anybody of you is having questions related to this topic, you may ask, sir, your difficulties. Sir, I could request you to please answer the questions. And sir, we definitely see which type of the medicinal plants the Kolam peoples are using to fight against this.
I think there are no questions. You can,
Okay.
of course, go ahead with probably. Yeah.
Okay. Thank you, sir. Thank you. Friends, I feel terribly blessed with the presence of our next guest speaker, Honorable Dr. Mumtaz Beg, Department of Integrative Biology, University of Guelph, Ontario, Canada. Without wasting, let me take this opportunity to introduce Dr. Mumtaz Beg. Dr. Mumtaz Beg is working as an Associate Professor, Department of Zoology, Government Vidarbha Institute of Science and Humanities, Amravati. And presently he is working as the Research Scholar, Department of Integrative Biology, University of Guelph, Ontario, Canada. Sir is expertised in the molecular biology techniques such as DNA, extraction of DNA and RNA, PCR, RT-PCR, and DNA sequencing. The molecular biology techniques. Sir is recipient of Dignity Postdoctorate Research Award in New York. He is also recipient of SDWA Postdoctorate Award in China. He is also the recipient of Phule Ambedkar Award, Government Vidarbha Institute for New York and Humanities, Amravati. He has conducted postdoctoral research work in many countries like America, Portugal, Germany, France, China, etc. He has published 23 research papers in the high impact transfers in many journals with citation index of 1085 out of which one paper is cited in Science Journal and two of the publications are cited in the reference book published by the Springer. So above all, I request Dr. Mumtaz Beg to please continue with his talk. Dr. Mumtaz, and two of the publications are cited in the reference book published by the Springer. So above all, I request Dr. Mumtaz Beg to please continue with his talk. Dr. Mumtaz, and two of the publications are cited in the reference book published by the Springer. So above all, I request Dr. Mumtaz Beg to please continue with his talk. Dr. Mumtaz, and two of the publications are cited in the reference book published by the Springer. So above all, I request Dr. Mumtaz Beg to please continue with his talk. Dr. Mumtaz, and two
talk. Let me clarify that I changed my topic talk to SARS-CoV challenges and promises, what it was being printed in the flyer, and I changed my this talk is to based on some promises, what it was new information that is being gathered recently about this and I changed my virus. This talk is, and I think based on the topic of your new information that is being gathered COVID impact on life sciences, and rather I would say virus, how, and I think the life based scientists, the topic of people in your life sciences or even beyond that will contribute to this challenge. Because this is not affecting the individual, it's affecting only individual across the world. So in this time, what is important, the combined effort is not very much required. Instead of individual going for a blame game. So in this talk, I mainly focused on some information which is being backed by pure science, going for a blame game. This information is being this talk I mainly focused on source from information which is a bulletin of Royal Society of Biology, to which I'm member since last 2 years. So I'm going to share this information which is being gathered by the Royal Society of Biology London, Royal Society of Biology. Also, I will share last 2 years. So our recently published study on this SARS-CoV. So let's move on to our discussion. Dear friends, one of the greatest challenge of this pandemic is how to counter the misinformation. Because there's a lot of information. If you make any search either on Google or challenge of this any social platform like how to WhatsApp or Facebook, there is a lot of tons of information. But all information is not true. It is very important that we must have access to the right information. And what is the definition of the right information? There is a lot of tons of information that is being backed by science. That is right. So being a access to the person right information and what is the related to life sciences or science, it's our duty to gather and disseminate this information. One of the tasks of work
I request all the participants to please mute their cell phones
right
so that we can be easily audible to Dr. Beksa. I request all the participants to please mute their cell phones so that we can be easily audible to Dr. Beksa.
News that really
I request all the participants to please mute their cell phones
about this pandemic
so that
and
we
there are
can
some
be
challenges
easily
too.
audible to Dr. Beksa.
In the coming years, we have to deal with this both promises.
I request all the participants to please mute their cell phones
One of the
so that we can be easily audible
very
to Dr. Beksa.
important challenge is to trace the origin of this virus. From where
I
this
request all the
COVID comes
participants to please mute their cell phones
COVID-19, commonly called as
so that we can be easily
zoonotic
audible
virus that
to
belongs
Dr.
to
Beksa.
important challenge is to trace the origin of a virus called as coronavirus.
I request all the
And
participants to
of
please mute their cell
animal origin.
phones
COVID-19, commonly called as
so that we
75%
can
of
be easily
infection
audible
virus that
to
belong
Dr.
to
Beksa.
or infection diseases in the last 20 years.
I request all the
Where
participants to
of
please mute their cell
animal
phones
origin. And you will surprised to know that
so that we can
60% of the
be easily
infection
audible
were
to
caused
Dr.
by
Beksa.
the virus belonging to this family of the last. This is not new.
I request all the
So
participants to
identification of
please
the
mute their
origin is
cell
very
phones
important. And equally important is that it doesn't always mean in science or in epidemiology. If some organism or if the disease is being detected at a particular place, it doesn't necessarily means that it started there only. Though it was first some trace in the Wuhan meat market, WHO has already constituted a team of international researchers to investigate further, though it was first, but it is not that easy. Trace in the Wuhan meat market, as I told earlier, this virus originated in the animal, so animal are the carrier. So identifying but it is not that easy. Animal, which is in the case of the SARS-CoV, suspected as bat is the flying mammals considered as the carrier of this virus. So identifying bat, which is in the case of the SARS-CoV is like finding mammals considered a needle in the haystack. ऐसे जैसा कि एक सुई को बहुत बड़े घास के ढेर में ढूंढना. So this is not that easy task. Second, now it's become a very ऐसे जैसा कि एक सुई को politically motivated and sensitive situation. So the US and China they started blaming. So it is not that easy, but already WHO has taken initiative and team is being considered. And the objective behind this investigation is that one and the review but available on the preprint server. The study was conducted by Australian researchers in collaboration with Spanish virologists. They analyzed the sewage water of some Spanish cities and they were surprised to find the traces of this SARS-CoV RNA in March 2019, much before the surprise when the outbreak was reported in the Wuhan in December 2019. So question arises from where this virus when the come in the Spanish sea witch before December 2019. So question arises from where this is really a challenging situation and come only the time will tell how the things move up. Already a team is being formed. Initially it was being carried out by only the time will tell how Chinese investigator and then the international team will join them. And hope the carried out by change of the presidency in US will improve this thing because science do not join them or science do not flourish much when people started blaming or blame game started. Let hope for the science do not join them or science do not. So another challenge to deal with this when pandemic is started, lack of transparency because many countries are facing this problem. Let hope for the and this lack of transparency is undermining the pandemic policy and many officials lack of transparency. They are because many countries are facing this problem and taking the place of an expert undermining the pandemic policy. As you may agree, you may agree with me that if one non-expert taking the start talking about things in which he or she do not have any expertise without consulting the expert, then it create a lot of problem and issues. So the same thing is happening with this pandemic also. Some officials they are making announcement and keeping the expert issues. So the same thing is happening. So that is not good with this pandemic also. So involvement of experts they are making announcement in the form of epidemiologies, keeping the expert the frontline worker, the scientists is very important to tackle this so involvement of disease. Another study has come out with a very alarming and interesting finding disease that there is a strong correlation between air pollution and COVID-19 deaths and where the air quality is very bad, the severity of this disease is very high. So researcher has clearly identified a clear link between COVID-19 and air pollution. And this we can also correlate with our capital New Delhi. Researcher has clearly identified thousands of COVID-19 positive patients are being emerging and many people are dying because the quality of the air is very bad in the New Delhi. So this is the one radiations are being emerging and many people are dying. Important challenge face this pandemic along with the air pollution. So this is the one radiations we have to deal with this important challenge thing dealing pandemic and also improving the quality of the air pollution. So many of the findings are also coming out and the sequencing of the genome has come out with some very fascinating findings. Many of the sequencing has revealed that there are many tiny variations which are found in the genome of this virus that can be exploited in the preparation of vaccine. Though these viruses term as SNP, single nucleotide polymorphism, but
this small variants, though this virus may responsible for varying degree of infection. We know that there are many reports coming that in some region this virus is more lethal and while it is not that lethal in other region. So why this is happening? So sequencing has revealed that there are some basic differences that contribute differently to the lethality of this virus. So why this happening? Very promising news which can be utilized in the preparation of very effective vaccine against this virus. So some other new testing finding are that beside affecting the pulmonary system, this virus also attack other some other important organ in the body like brain and cause delirium, which is a kind of loss of memory attack. Because long it was speculated that this virus not only affect the pulmonary system but also affect other organ of the organism. Because this family of it was speculated that coronavirus also cause the many deaths So when we plotted this graph, we find some very diverse and very different information. So for Coulson constant, what we found that starting January, the infection reached its peak at around mid of the February. You can see here in the, and after mid of February, the curve flattens or it reaches the plateau. Okay, so all is well here after mid February. But when we use the second model, Coulson's exponential, the things are very drastic and very different from model A. There is a continuous increase. So you can say that in other words the growth is skyrocketing. So this is very alarming situation. And also if you see the current scenario, the number of positive is he or she do not have any expertise. Without consulting the expert, then it create a lot of problem and issues. So the same thing is happening with this pandemic also. Some officials, they are making announcement keeping the expert backdoor. So that is not a good sign. So involvement of expert in the form of epidemiologists, the social worker, the frontline worker, the scientist is very important to tackle this disease. So another study has come out with a very alarming and interesting finding that there is a strong correlation between air pollution and COVID-19 death. And where the air quality is very bad, the severity of this disease is very high. So when we plotted this, researcher has clearly identified a very clear link between COVID-19 and air pollution. And this we can also correlate with our capital New Delhi. Daily thousands of COVID positive patients are being emerging and many people are dying because the quality of the air is very bad in the New Delhi. So this is the one very important challenge we have to face this pandemic along with the air pollution. So we have to deal with this twofold thing, dealing pandemic and also improving the quality of the air. So many of the findings are also coming out, and the sequencing of the genome has come out with some very fascinating finding. Sequencing has revealed that there are many tiny variations which are found in the genome of this virus, that can be exploited in the preparation of vaccine issues. Though these viruses, the same thing is happening. Variations are very tiny with this pandemic also in technical term as SNP, single nucleotide polymorphism. But these small variants may backdoor responsible for that is not a good varying degree sign of infection. Expert we know that there are many reports coming that in some region this virus is more lethal, and while it is not that lethal in other region. So why another happening? So sequencing has revealed that there are some base pair differences that contribute differently to the lethality of this virus. Strong correlation between this air pollution and very promising news, and which can be utilized in the preparation of very effective vaccine against this virus. Disease is very high. So some other interesting findings are that beside affecting the pulmonary system, this virus also attack other important organ of the body like brain and cause delirium, which is a kind of loss of memory, and many people are dying because long it was speculated that this virus not only affect the pulmonary system but also affect other organ of the organism, because this family of coronavirus pandemic also cause the many gastrointestinal diseases in many domestic animal like cat, dogs, and even cattle. So many of the findings, some of the coming out scar this infection they leave if somebody contact the disease. So one should not take this virus very lightly. Sequencing has revealed that there are many. Also, there are some promising news that this virus that can be exploited in the infections are very rare in newborn issues. Though these viruses, because most of the infections variations are very tiny, are reported in this adult also in human technical term. This is good news that newborns they are not that susceptible to this virus as compared to young and old age people. So this is based on the study that has been done in UK, and where newborn were analyzed for the virus is more this virus, and it was found that this COVID infection is very less in the newborn babies. Another study sequencing. So another good news is that differences that schools are probably not the hotspot of COVID. So there are some studies promising that is being carried out in many which can be countries where students and teachers were effective vaccine against this virus analyzed for some other the presence of this virus, and it was found that this virus beside affecting found in the pulmonary system this adult students compared to younger one. So in adult students of the body like above the age of 14 they are more prone to develop COVID infection compared to less than 14. So because long it was speculated that good news that this virus schools are not only affect the turning to be a system but also hotspot of this organ of infection. That's why in many countries people are because thinking to reopen these schools. Already in Canada the schools are being reopened, although there are some gastroenteritis surge in many of the cities. Recently, the Toronto has undergone 4 weeks of lockdown, but dogs and schools they are operating in different mode. They are not allowing all the students to come together. They are divided into batches, and there is no sharing of food in the cafeteria. So this is a good news regarding this virus that students sorry school is not the hotspot for this virus. Rare in newborn issues. So another promising study revealed that those who affected with this virus they develop good immunity against this virus, and this lasts for years as compared to. So this is based on a new data which people. So this is based on the studies that has been finding that UK immunity developed against this virus do not lost very. It was found that soon and COVID infection, the person who acquired this infection get immune, develop resistance for this virus for a longer time. So this is a good thing to develop a very effective vaccine and protect against this virus. So another important good news about this analyze coronavirus is that some already many countries they are in the process of developing vaccine against this virus. Our country India is also one of the leader in this process. So the most good thing about this vaccine development is that already there are many vaccines are turning to the market for coronaviruses in the veterinary medicine. As I told earlier, coronavirus is a very rare family of viruses and it causes many diseases, including this flu and recently the Toronto has gastroenteritis in livestock, especially cattle, dogs, and other farm animals, including pets like dogs and lions. So there are already bunch of vaccines which are developed against this virus. So that experience and expertise can be utilize this is the development of vaccine against this virus in human also. So this is a very promising field and news too. So what we contributed in this SARS-CoV is that we analyzed some 250 genome which were deposited and sequenced by Indian laboratories, and we use most prudent methods of genomics tool. I collaborated with my national collaborator from Hyderabad and use the high computational facility of Toronto supercomputer, and I'm very happy to say that this paper is being accepted for publication in one very prestigious journal published by UK called Future Virology. This is the proof of the same vaccine paper, which hopefully is published at the end of this month or start of the next month. And you can find the preprint of this paper online on Matrix, and all the research related to COVID is made free as per the new guidelines of WHO process. So in this study, we analyzed some 29,000 thing about this vaccine base pair. Already there are many vaccines genome of this SARS-CoV in sample across India, and we have some very interesting findings which I would like to share with you in the next slide. So this is the phylogenomic network we built using 286 whole genome of the SARS-CoV live virus, especially cattle, deposited from various laboratories across India. You can see the farm animal color code. Yellow is the sign here, and show the virus originating from the Wuhan province. This fluorescent color, blue indicate the Gujarat, orange Telangana, Delhi, Tamil Nadu, promising. So what is important about this phylogenomic network is that you can find two clusters, A and B, and these two clusters are the important cluster in the engine isolates, and all these are very much connected. The number which can tool you can see in the red like 1, 2, 3, 5. They are the mutations, number of mutations, high computational facility of. It is very interesting to see that supercomputer number of mutations which I'm very happy to added to this virus. So we cannot say that this virus is not mutated, and it is very important to understand this mutation and mutation rate at which this is the virus is mutating. If we very faithfully and very robustly we record all these mutations, then only we can very robust vaccine against this. We can find the treatment of this. So online this understanding of the variation in the genome is all important to have very effective vaccine as per the against these new guidelines of WHO.
So after
So
building
in this study,
phylogenetic network
we
to understand the
analyze
relationship between
some
all these
good
virus
thing about
isolates,
this
our next step
vaccine
was
is
to
that
understand how this virus
genome of
is
this
actually
SARS-CoV
or this infection is progressing
sample
in the real time.
across
I mean,
some very interesting
what is
findings
the
which I
present, past, and the future
in the next one.
trajectory
Cause
of this
it is
pandemic
the
in the
phylogenomic
countries of
network we build
India.
using
So for this purpose,
286
we use some very
whole genome
hardcore
of this
sequencing
SARS-CoV livestock,
analysis,
especially cattle,
and we use
deposited
two
from
models
various
here.
laboratories
One
across
model
the India. You can
coalescence
see the
constant, we
animal
call it
color
as
code. Yellow is the
A
sign
here,
here, and the show
the
the
plot, and
virus
another model we call
mutating from the Wuhan province
coalescence
actualized in the
exponential. It is
development of fluorescent colors.
illustrated here
This virus now
as
indicated
B. So
Gujarat,
constant means that assuming
Telangana,
that the
Delhi,
population
Tamil
is
Nadu,
constant, of
so
these viruses,
what
and
is
B,
find two clusters A and B,
growth or the infection is increasing at
and these two
exponential rate.
clusters are the
So when we
important
plotted
cluster
this
in the Indian isolates,
graph, we find some very
and all
diverse and very
these
different
are
information.
very much connected
So
with different methods. The number
for
which can
coalescence
tool
constant,
you can
what we
see here
found
in
that
the red, like
starting
1, 2, 3,
January,
5. They are the mutations,
the
number of
infection
mutations,
reached its
high
peak
computational
at
facility of. It is very
around
interesting to see
mid of
that
the
supercomputer number
February.
of mutations
You
which
can see here.
I'm very happy to added to
And
this
after
virus.
mid of
So we
February,
cannot say
the
that this
curve
virus is not
flattens.
mutated,
It reaches the plateau.
and it is very important to
All is well
understand this mutation
after
and mutation rate at which
but
this is the virus is
when we
mutating.
use
If you
the second model,
very
coalescence
faithfully
exponential,
and
the things
very
are
robustly
very
we
drastic.
record
In other words, the
understanding of
growth
the
is skyrocketing.
variation in the genome is
So
all important
this is
to
very
have
alarming
very
situation.
effective vaccine
And
against these
also, if
new
we see
guidelines
the
of WHO.
current
So
scenario,
in
the
this
number of
study,
network
we
to understand the
analyze
relationship
two
between all
communities
these in our
virus
virus
thing about
isolatry
this
also. Our next step was
vaccine
to understand. That's why this virus
genome of
need to be cautious,
SARS-CoV
or this
and
infection will not take progressing
sample
for the time granted.
across
So based on this, our so what is the projection? Present, past, and the future
now this is the
trajectory
time to
of this
thank
pandemic
all
in the
contributed
countries of
to
India.
this research.
So for this
Example to my
purpose, we
collaborator,
use some
Dr.
very
Ravi Kumar Tandon,
hardcore
who is scientist at National
analysis,
Animal Biotechnology Institute,
and we use two models here. One
Hyderabad,
model,
India. My student,
coalescence constant,
Samir
we
Afara,
call it as
another student, Dr. Ashwinat Kumar, and my mentor here, Elizabeth Bolding, for providing me an access to state-of-the-art high
another
computation
model
facility
we call it
of Toronto. Wuhan province
coalescence
actualized
exponential.
into fluorescent color staff members of
Illustrated here
NIT,
as B.
Rajkamal,
So
Gujarat,
constant means that assuming
Telangana,
that the
Delhi,
population
Bharati
is
Arts, Commerce and Science College, Arni, Vatmal, my student Rupali, and all those who put their efforts in this conference. last but not least, I'm also thankful
What
to
is
all audience for A and B.
Go for the receptionist.
Thank you very much. And if you have any query, you can
Very important graph
have it provided on the slide.
famous and very
At the end,
information.
I would like to conclude that this is a very critical time we all are
What we
facing,
found that
and
start
we can fight
January, the infection
to beat this
peaked at
together.
around mid of the February by 20. And after mid of February, the curve flattened.
Policy constant Sapara, the student operational at Goa, and my mentor here, Elizabeth Bolding, for providing me an access to the Sudar High Computation Facility from Toronto.
This was Dr. Mumtaz Beg, sir. I thank you, sir, for sharing your knowledge and research work in form of the video with us. Hope we will get another chance to listen you in future. Thank you, sir. If you have I request all the participants that if you have certain queries regarding this topic, that is SARS-CoV-2 challenges and promises. If you have certain queries or questions, you can personally mail Dr. Mumtaz Beg sir on his mail ID, which is as shown in his last slide. Once again, I thank you, sir. Now it's my pleasure, friends, to let you know that Dr. S.R. Yadav, Senior Scientist, INSA, Shivaji University, Kolhapur, is our next speaker. May I request Dr. P.J. Aute, Head Department of Zoology, to give the introduction of Dr. S.R. Yadav. Over to you, Dr. Aute. Due to some technical issue, I request to just have the few minutes of 5 minutes break. We'll again resume and start further after 5 minutes. Thank you.
किती असेल? दोन घेऊन गेलो. तर तिसरे सर्वात
May I have your attention, please? The feedback link will be sent on the Telegram group. If any of you who have not joined the group, the email is sent to you. Please go through that and join the Telegram group so that it will be better for you to send the feedback link. Thank you. I welcome you all. Friends, it's my pleasure to let you know that Dr. S.R. Yadav, Senior Scientist, INSA, Shivaji University, Kolhapur, is our next guest speaker. May I now request Dr. P.J. Avte, Head, Department of Zoology, to give introduction or introduce briefly Dr. S.R. Yadav. Over to you, Dr. Avte.
Thank you very much, madam. Good afternoon. I extend warm welcome to all dignitaries and participants. I find immense pleasure in introducing our chief guest, Dr. S.R. Yadav sir, who has been worked who has been working as senior scientist at Indian National Science Academy, in short INSA, of Shivaji University, Kolhapur. Dr. Yadav is an outstanding plant taxonomist of international repute, an excellent teacher whose floristic and taxonomic works, particularly flora of Kolhapur and grasses of Maharashtra, have tremendously influenced many students to take up plant taxonomy as their career. During the last 3 decades, he not only recorded a new flowering plants, Hamili Hydatelaceae, but also discovered 74 species new to science. He is an ardent field botanist and outstanding plant photographer. He has completed over 20 projects. He has significantly contributed to establish lead botanical garden of Botany, Shivaji University, Kolhapur. He has vast administrative experience. He has published 270 research papers in national and international journals. Sir has guided 11 students for MPhil and 26 students for PhD. Sir has organized 2 international and 4 national conferences. He has attended more than 50 national and international conferences and delivered keynote invited lead lectures. He is engaged in plant explanation exploration of Western Ghats, biosystematics and conservation of rare plants. His special interest is an Aponobotaceae and bulbous monocots. Now I request Dr. S.R. Yadav sir to start his presentation.
Is it visible? Hello?
Yes, sir, visible. Visible and audible.
Okay. So I have to do it. Presentation mode. So full screen is available? Hello?
Yes, sir.
Full screen is seen?
Yeah.
Okay. So good morning, good noon to everyone. It is my great pleasure to talk to all the people in the age of COVID-19. You know, COVID-19, for so many months we are blocked, and one lecture I gave in one college, and same lecture I will be delivering today also. I am thankful to organizer. In the beginning only I want to say, because I forget when I go for lecture and speak, and finally I forget. So for nice introduction and inviting me for sharing my thoughts about this one, COVID-19. Before that, my lecture is on angiosperm diversity of India and antiviral herbs in protection from COVID-19. Our country is known for Ayurveda, known for Siddha, and I know right from my childhood, whenever I get some fever, some kada is given, some decoction is given, and in 1 or 2 hours, I get relief from that temperature which we get. These are simple things now. And COVID-19 is also a virus by which I mean is a virus which has spread throughout the world and the entire world is suffering. But in India, we have many antiviral plants and we are very rich in biodiversity. So I will show initially what is the biodiversity of Western Ghats so that you will enjoy some of the plants. And then I will suggest the plants which can be used for, facing COVID-19. Now, this graph, last time only I have shown this graph, how this COVID-19 patients have increased. And there is a possibility of second wave of COVID-19. And some people have written it could be tsunami. So many people may get infected with COVID-19. So all of us, we have to be alert and we have to follow instructions of our Honorable Prime Minister that keep distance, mask your face, your mouth and now nose, and don't go to crowded places. So these are the solutions. We have to protect ourselves. That is one. And another thing, in case if we get infection, what we can do. But before that, also our Ayurveda has told prevention of diseases is the best way. And only if we get infected, we should go for medicines. Now, what is our country? Biogeography of India. This is what I want to tell you. What are the noteworthy features of our country? It is the 7th largest country in the world. And moreover, it is the 2nd largest nation in Asia with an area of about 287,000 square kilometers. Now Himalayan ranges we have, which is up to height, altitude is up to 8,586. And it measures 3,214 kilometers from Kashmir to Kanyakumari, and from Gujarat to Assam, it is about 2,933 kilometers. Land frontier is about 15,200 kilometers. Coastline is of 7,516 kilometers. So it is a very large country in South Asian countries. Encompasses varied landscapes from Himalaya peaks to the Indian coastal line and wide variety of physical and climatic conditions. India has great diversity of habitats, climate wise, soil wise, temperature wise, rainfall wise. We have a lot of diversity in climatic and physical conditions of our country, and harbors varied ecosystems like tropical rainforest to high alpine cold deserts, grasslands, wetlands, and coasts. It embraces 3 major biological realms like Indo-Malaysian, Eurasian, and Afro-tropical. And it sustains and supports immense biodiversity. Today we have more than 1.25 lakhs of species in India, which includes both microbes, animals, and plants. It has 10 biogeographic zones and 26 biotic provinces. Why I am telling this, especially for students, that we have great diversity in climate and in physical factors. Here you read this sentence, 2.5% of Earth land area. It has just 2.5% of Earth's area, but supports 8% of recorded species of the world. 8%. So we have very rich biodiversity. We have millions of races, subspecies, and local variants of species, and the ecological processes and cycles that link the organisms into populations, communities, and all different ecosystems. It is the, but this point, it is the country which is the second largest populated country in the world. Though we have great biodiversity, we have great number of people also in the country. And that there is a great pressure on our natural resources. A majority of its population directly, majority of population depends on biological resources for livelihood. So these 2 points are of critical importance, because every day we are losing a lot of biodiversity from our country, from our mother nature. So this is I already these are the biogeographic zones of our country. I will not go to these things, but great diversity in physical and climatic factors. We see we have Trans-Himalaya, Himalaya, desert, semi-arid, Western Ghats, Deccan Peninsula, Gangetic Plains, coast, northeast islands, and so on. So this way we have 10 biogeographic zones. Agroclimatic zones of India, I don't know whether you think anytime in your life that we are the richest people on this planet. Why I am saying, reason is we have so much diversity in food. Our food basket is very, very rich. And how many things we eat per day. It is not with developed countries or Europe or America, where they just bread, beef, and some fruits. But here we have great diversity of agricultural products. So we have Western Himalaya, we have Eastern Himalaya, Lower Gangetic Plains, Middle Gangetic Plains, Upper Gangetic Plains, Trans-Gangetic Plains. All these, there are about 15 agroclimatic zones in our country. Now, one of the 18 mega diversity countries already I have told you, and these points I have already said. Now, biodiversity hotspots. Students should know what are the biodiversity hotspots, why they are recognized, and what is their role in conservation of biodiversity. So about 36 hotspots have been recognized throughout the world, and they are the areas which have more rich biodiversity and high endemism. So that way, these are the biodiversity in our country. We have 3 biodiversity hotspots. Right now, you see every year how many species are described. So about 20,000, 2,000 species of vascular plants are described per year. 2,000 new species are described from plant sciences. And by this rate, if we go on documenting species, it will take long time to document our species. You see these species, okay. Then biodiversity hotspots in India, we have these Western Ghats, and these Western Ghats play a very important role in our life. Then this Ceylon part, Northeast India, Himalaya, Western Himalaya, these are the areas which are with very great biodiversity. So range of biodiversity is the total variety of life on Earth. It includes all genes, species and ecosystems and the ecological processes of which they are part. Ecosystem diversity, species diversity, genetic diversity. So these are the important things. And one should know, and we count biodiversity in the number of species. So currency to measure biodiversity species, below that we have genetic diversity, so many genes in each species, and then above we have ecosystem diversity. So that way diversities can be studied in under three heads. Now here you see what is the biodiversity of your country as compared to the world. Now, for each group I will not go. I will talk only about angiosperms, because which is a very important group for all of us. So today, total families recognized by APG4, angiosperm phylogeny group, fourth classification is 416 families, of which 251 families, members of these families are found in India. Then total number of species described so far is 2,95,383, belonging to 13,164 genera. In India, we have 18,259 species belonging to 2,988 genera. So almost percentage of Indian flora is 6.18, and that way we have very rich biodiversity of angios. Similar is the case with pteridophytes, bryophytes, algae, lichens, fungi, and we are very rich. So today in India, if you ask me how many plant species are found, it goes to 48,158 species. Of course, plus and minus, there will be figures will be different in different this one. So if you see endemism, all of you must be now knowing plants which are restricted in distribution, they are only found in those areas, nowhere else in the world. So endemism in Indian flora, again I will say only about angiosperms. Angiosperms, we have total species 18,259, of which 4,303 species are exclusively found in India only, nowhere else in the world. And these endemic species are very specific in their requirement. They are restricted in distribution, and majority of them are critically endangered. So that way, angiosperms, similarly is gymnosperms, pteridophytes, bryophytes, all these figures I have given, you can read. I need not to read it again. Right? Mega and micro centers of endemism. Now see, these are the greenish color areas. These are the mega centers of endemism, where we get high endemism. So these are the areas: Western Ghats, Western Himalaya, and Northeast India. Okay, Arunachal Pradesh and this Sikkim, Himalaya, all these are very rich. And Andaman Nicobar Islands. And Nair has recognized, Sanjapa has, micro centers, 25 micro centers of endemism we have recognized in India. See this different life forms in endemic species. Herbs are 53, then shrubs, trees, climbers, undershrubs, and bamboos. So there is a great endemism in our country. So family poaceae, grasses, I don't know, all of you should think that grasses are like our Rusi Munis who work for society, and our 80% of human requirements, 80% 80% requirements of human beings come from poaceae, come from grasses, our food, our fodder, our meat, our sugar, and whatnot that comes from grasses. In addition to that, they play a major role. And in your country, we have highest number of endemism in poaceae. So 355 species are endemic to India. Then Orchidaceae 274, Leguminosae 274, Rubiaceae. So this sequence you can see: Poaceae, Orchidaceae, Leguminosae, Rubiaceae, Acanthaceae, Asteraceae, Balsaminaceae. These are the major families in which high endemism is seen. And you know, Balsaminaceae, balsam, terada, okay, Gaurichi Phula we call it as, we have more than 169 species of balsams. And in our Western Ghat, we have about 86 species of balsams. Almost all of them, I mean to say 82 species are endemic to Western Ghats. How many of us we know these balsams? Okay, so Lauraceae, Asclepidaceae, all these are families with high endemism. So number of endemic angiosperms restricted in different phytogeographical regions. Western Ghats, you see, in Western Ghats, we have 2,116 species of endemic to Western Ghats and our country. Then Eastern Himalaya, 416, Northeast region, 395, Western Himalaya, 297, Deccan Peninsula, 312, Islands 278, then West Eastern Ghat 166. So these numbers, you know, of endemic taxa in different regions is shown here, which will be useful to students who are doing research in botany. Right. Then Leguminosae has highest number of endemics. Poaceae has, in dicot we have Leguminosae, in monocot we have Poaceae with higher number of endemism. Okay, so these are the families like Rubiaceae, Acanthaceae, Asteraceae, Balsaminaceae, which have very high number of endemism, similarly in monocots. Now, that is some preliminary information I have given about our country, which is very rich in biodiversity with high endemism, and that's why one should know what is our strength. Unless you know the strength, your strength, you cannot perform better. Now I come to Western Ghats. It is an important biogeographical province of India. Now you see, this is a narrow strip right from river Tapi to Kanyakumari. This is that narrow strip of mountain ranges which harbors great biodiversity. It plays a very important role in hydrological cycle of peninsular India. This entire peninsular India depends for water on this Western Ghats. Okay, it plays important in a hydrological cycle of the peninsular India. Because they are there, we have perennial rivers, and all those rivers which flow east irrigate entire peninsular India. So you can understand importance of Western Ghats. Climatology, good climate which we enjoy in peninsular India. Western Ghats govern our climate, and very importantly, I want to tell you, there are more than 12,000 species of plants in Western Ghats. It occupies just 5% of our India's area, this small strip, 5% of India's total area, but supports 26 to 27% of flowering plants. So Western Ghat is a treasure of biodiversity, treasure of angiospermic plants. Unfortunately, most of the developments are done in such areas which are responsible for loss of biodiversity. High endemism, we have, about 2,253 species endemic to Western Ghats, and 1,273 are exclusively found in Western Ghats only. So that way it has its significance in our life. Now, I want to tell you see this, some Western Ghats, some of the Western Ghats, see this Western Ghats, right? Now, these Western Ghats, which harbor that biodiversity, which I have already said, and see these mountains, waterfalls, and such a diversity in your habitats. We get great diversity in habitats and great number of species. Now you see these mountains, if you want to search for endemic plants, they are found in these areas. And see this Western Ghats, Khas Plateau. I am showing you just to show you what you are, what is our country. Himalaya I am not showing, it is a Western Ghat which is equally rich as Himalaya. And see this Khas Plateau flooded with so many plants, herbaceous plants. In summer, these are barren rocks. In rainy season, we have wonderful wealth of flowers, floristic wealth of Khas Plateau, right? Now you see this Khas Plateau, which is flooded with Senecio grahami flowers. Entire plateau has become golden yellow, and you can see this, impatiens which are growing, and we have many species of impatiens. Then see this close up. See these utricularias, eriocolans, impatiens, then comelinas, murdanias. All that is a wonderful land of flowers. One should go and see. And these bioresources, right? These bioresources are of great significance to all of us. See this. Now I showed some plateaus. I am showing these flowers. All these are flowers from Satara, Sangli, Kolhapur, Sindhudurga district. See this diversity of flowers. Why I am showing that, biologists should understand they are the people who have to play great role in our betterment of mother planet and welfare of human beings. See these flowers, they are so diversified, so diversified. See this Anonasi members, what a structure each one is having. Each one is so different, and anybody should get really impressed by the diversity created by nature in plants and animals. Animals, equally interesting diversity we get. Plants, we get different diversity. See these flowers of Murdanias. Flowers of Murdanias. See this beauty. See these fertile stamens, and 3 stamens have modified into staminodes. They have fluorescence, and all that, evolution, which we study in classes, is a very unique, very fascinating for anyone. We are finally trying to understand evolution of different organisms on this planet. From that angle also, our biodiversity is unique. See these eriocolans. In eriocolans, I described some species about 15, 20 years before, 2-3 species. After that, our students have added more than 40 species of eriocolans, and today we have 115 species of eriocolans in India. And students are doing wonderful work in documentation. And all these eriocolans, today we don't use any eriocolan, but see the diversity of eriocolans and micro diversity one must learn to study, right? See these flowers of different families. I will not give all the names. It is you to know the names. I can tell name of every plant, but see, I will not tell it. See this diversity. Legumes, Commelinaceae, Boraginaceae, Gesneriaceae, Tiliaceae, and so on. All that diversity. It is an amazing diversity in India. See these legumes. Legumes. Go close to the flower and there is a heaven. You to understand. See this diversity, Smithia flowers, then our Macrotilia, Alicicarpus flowers, all that is a wonderful diversity. Why I am showing this diversity? That this diversity only is going to help us in overcoming many diseases, not only COVID. They have wonderful phytochemicals which can be used for various purposes. See this Abituna ranadi. It is a Songanta, beautiful flowers, and it is a critically endangered species. Again, we have to save. See these Vignas, Vigna conitifolia, Vigna radiata, Vigna mungo, Vigna sayadrika. So many species are there. 30 species of Vigna are found in India. And from India, 3 crops have been developed in genus Vigna. We are rich in that. See this Clitoria, then our this one Ichinomini, Alicicarpus, all these wonderful diversity of our country. Okay. See this legumes only I am showing. Now Cisalpinaceae, you see this diversity of flowers. Do you find 2 flowers exactly similar? Answer is no. So such a tremendous diversity has been created by nature. Mimosaceae, so much diversity. See this Ereucolan. Don't you get impressed by this, our diversity? And that's why I said you are so lucky that you are in a country which is very rich in biodiversity. And if there is a true capital of human being, it is only biological diversity. Your aeroplanes, your cars, your motorbikes, your bungalows, they are secondary. Primary is the biological materials which we need, which fulfill our basic needs, and that way, biological diversity or biodiversity is a biological capital of human being, which is most needed by human beings. All other things, if they are not there, no problem. We can live happily. But if these biological materials are not there, we cannot live. So I will come to that point. See this, okay, this biodiversity. And to study biodiversity, we need taxonomists. And unfortunately, taxonomy is considered to be very old science. There are no jobs and people run away from taxonomy. I mean, people think that taxonomy has no scope because there are no job potentials. But dear students, we learn anything not to just feed stomach. We have many other functions to perform, especially, welfare of human being, welfare of mankind and Mother Earth. And for that, we need taxonomies. So taxonomy is a very important branch of biology, which is mother branch of biology, which nurtures every branch of biology. So see this diversity. And, we have CBD, Convention on Biological Diversity, technology transfer, information, exchange of scientific and technical cooperation, equitable sharing, all that, these biological materials, which are very important. Convention on Biological Diversity, which came into existence to protect these plants, to utilize these plants sustainably. And now you tell me, this is the diversity of flowers which I have shown. Just I have shown diversity of flowers. I can show you diversity of fruits, diversity of roots, but diversity of leaves, diversity of stems, Such a diversity, I feel very happy that I took botany as my career subject, right? So this is a diversity which I want to show you. See these arissimas, what a wonderful structures, their arrangement, their symmetry, all that is amazing to me. These are the Amorphophilus species from our country, from only peninsular India, not from entire. I know. Then I come to my lecture topic, herbs and human health. See this human health, herbs, these sadhubuas, rusimunis, vaidyas, which sit and sell these medicines. All medicines, at least in the beginning, have come from plants. So health and human health, herbs and human beings, there is a great correlation. We are today surviving, or we are safe from many diseases because our plants have synthesized many phytochemicals which we have used to control our diseases, right? Now, I want to tell you, I come here, Mahasudarshan Kada. I am showing you, all of you know, Sandu prepares Mahasudarshan Kada and Jhendu prepares Mahasudarshan Kada. Today we are in the age of COVID-19. All of us, we are with afraid mind, with suppressed mind, under psychological pressures. We are not able to meet so openly. We are not able to greet. And all that is created by this COVID-19. Can we keep COVID-19 away from us? Are there measures for prevention of COVID-19? I must tell you, I am so happy. In other countries, the death rate is very, very high, very high. But in India, death rate due to COVID-19 is comparatively very low. People suffering is very low. Why? What is the reason? Because we are more resistant and we have our lifestyle, because of which we are more resistant. And we have our Ayurveda, Siddha, which helps us to keep us healthy. Our immunity is good, and that's why we are able to do it. Now this Mahasudarshan Kada, I am here as a teacher, not to advertise anything about anybody, any company. But Mahasudarshan Kada, all of us, we are botanist, zoologist, Ayurvedic doctor. What is there exactly, which I wanted to know after COVID-19? You see this Mahasudarshan Kada includes halad, kurkuma longa, daru halad, Berberis aristata, moti ringni, Eclipta prostata, Piper longum, Clematis heni, Tinospora cordifolia, Funaria indica, Zygophyllum arabicum, Picrorhiza curria, Gentiana curro, all these. So in Mahasudarshan Kada of Sandu, we have 54 plant species which are used, and that decoction is given. Now, if you take survey of all those plants, about 50% plants which are used as antiviral plants. Most of them are antiviral plants. And that way, this is Mahasudarshan Kada by Vaidyanath. Here also there are 55 ingredients and almost all of them are plants, and most of the plants, all of you know, many of them are known to you. You can go through this list. By showing this list, I want to tell you the Mahasudarshan Kada, those who take regularly, they have very, what we call, immunity, and viruses don't attack to them. So that way, I also take these kadas in the night for last, say, since COVID, I am taking, and so far at least, I am not infected by this COVID-19. Now, then another, Indian plants with antiviral properties. All of you, botany zoologist people should know, and botany students especially should try to learn something from COVID-19 and create new opportunities for themselves. Now, what are the plants which are of antiviral, which have antiviral properties? So English name, local name I have given Allium sativa. Our lasun. Whenever we have temperature, we make mala of that, this one, what we call this, lasuna mala, and put it as a mala. Why we put it? Because they are antiviral. So Allium sativum, Alae barbarensis, Alstonia fininata, Amaranthus tricolor, Annona reticulata, Androdendron paniculatum, Ajada richta indica, Bacopa monieri. All these plants, if you see, majority of them are antiviral. So see, Allium sativa is useful medicinal use, mills, poliomyelitis, herpes. I don't know these diseases, but on these diseases they are used, and one should now use this knowledge, our traditional knowledge. India's traditional knowledge has a great significance. See this long list of plants which have antiviral properties. Jaundice, jaundice and stomachache, and this one, all these eclipta, prostata, glycosmis. This list I am providing, this presentation will be given to you by a organizer, and you can go through this list. I need not to go through this list. Right? So see this phytochemicals which are found in this, which are used active against viruses. All this data is essential, and that way antiviral plants I have introduced to you, and we can use it against these fevers or against this COVID-19. Now, from local doctor, I asked some doctors, and what are the plants which are used for as antiviral plants? Now, clematis, Morwell, clematis gauriana, we call it as, Berberis aculeata, Sorcia chiraita, Andrographis paniculata, Hemidesmus indicus, Allium sativum, Curcugo longa, Gingiber officinalis. All these plants, one doctor told, which they use to control antiviral, against viral fevers. Now, all these are components of Mahasudarshan Kada. Here, if you see another list, Acalipa indica, Acanthus illicifolius, Cinnamonum gelanicum, Ailanthus triphysa, Centipeda minima, Clerodendron inermi, Crisa critica, Crosandra infundibulifoli, Trichosanthus, Todalia asiatica, Sorcia densiflora, Stribulus. All these plants are used by doctors for treating patients with virus infection. And now this information we should collect from all over the country and compile together. So mechanisms of action of existing antiviral plant drugs to prevent viral entry into the cell. Adsorption of the virus has to be avoided by antibodies. Antibodies or specific ligands. Inhibition of viral uncoating after endocytosis by caspid stabilizing agents and blocking of endosomal ion channels. DNA and RNA replication can be suppressed by inhibition of DNA or RNA polymerase by endonucleases or by nucleoside analogues. Virus replication can be interfere with nucleotide analog. Okay, so these substances are peptides that block protease substances. This leads to suppression of maturation and interruption of viral replication cycles. I am not an expert. From some reference, I have taken it, how our plants work in controlling viruses. and increasing our immunity. That is the mechanism of antiviral plant drugs. Now see this Andrographis paniculata, which grows everywhere, right? Andrographis paniculata, which grows everywhere. You know, one of the director of Directorate of Medicinal Plants, CSIR, he always puts one leaf of this plant in his drinking water in the morning and takes that water for whole day. He never gets viral infection. These are the simple things we underestimate. We to treat one COVID patient, now people charge 3 lakhs, 4 lakhs, 5 lakhs, but the medicine which is in your around your house we don't use, which does not need money. So Andrographis paniculata, which has andrographolide, is a major compound in terms of bioactive properties and is in abundance. So we can control virus infections. Many of the andrographoids analogues indicating immunostimulatory properties and show impressive antiviral properties. Now, why our students should learn these things which are of great practical value to everyone, and we can help our society with our knowledge. But unfortunately, doing botany only for getting job and getting bread and butter. It is not a purpose. Another plant equally effective is Inicostema axillare, which grows all over Maharashtra, and you see how it looks. It is a close relative. It is again from Jansenaceae, from which Sorcia chiraita comes. The leaf extract is used as an antiviral in many skin diseases and viral infections. Now, why I am showing these plants, we should help. Now, this is a Sorcia densiflora, which grows in Panchgani Plateau and hills around Mahabaleshwar. And this is a substitute used for Sorcia chiraita, which is a Himalayan plant, chiraith, which is a well-known medicine. The bitterest compound which we find is in Sorcia chiraita. Similarly, Sorcia densiflora, which is our plant, is also is used as an antiviral plant like Sorcia chiraita, rich source of bioactive compounds like terpenoid, phenolics, and antioxidants. Right? Now, how many Sorcia species are found in Western Ghats? As per my knowledge goes, there are more than 8-10 species of Sorcia in Western Ghats in different parts. They have almost similar compounds, less or more, but they can be used as a substitute for Sorcia chiraita. And see this Tinospora cordifolia. This is queen of medicine, I will say. Reason is Tinospora has lot of importance. So it is a very, very important plant of our country, which has antiviral infections. And it is an antiviral and many diseases. It is useful. Tinospora. Why can't we? All in all villages on hedges, this plant grows. Tinospora cordifolia is rich in tannins, alkaloids, cardiac glycosides, saponins, triterpenoids, phytosteroids, and phytophenols, which have proved to be of great medicinal value and act as immune modulators, anti-diabetic medicine, anti-allergic, anti-leprotic, and anti-inflammatory agent. What else you want? Such a wonderful plant is growing around you. It is like our mother, which cares if you use it for your health. Recent research was conducted and a remarkable activity has been proved on the activity of Tinospora cordifolia extract on HIV. HIV is also a virus. Also, few studies have been conducted on the antiviral properties of Tinospora and have been reported to be of great value against Herpes virus. See, these are the things with us. Now another Ancestocladus henianus. This is a plant which grows in Western Ghats and it is endemic to Western Ghat. It is good source of botulinic acid, which is used against many viral diseases. It is for HIV, it is for many other diseases. The plant has antiviral, antimalarial, and neoplastic-like activities. Plant is used to treat AIDS patients, right? So just I am showing you our plant wealth. This is Quansium finistratum, which is rare plant, endemic plant, especially found in Kerala. But this Quansium finistratum is used by many tribal people as a source of anti-diabetic, anti-tumor, and antimicrobial drugs. So we have wonderful herbs with us. Only we should know from our tribal people, from our local people, that is we call traditional knowledge, which we have to gather. Every botany student, every zoology student should gather this treasure of knowledge and make it available to scientific world. Vidania somnifera, I need not to tell you. Vidania ashwagandha compound Vidanon helps to block or weaken the entry of COVID-19 virus. This is a reference, Balakrishna et al. 2020. So if you are taking ashwagandha powder, no COVID-19 virus will enter in your body because it blocks or weakens the entry of COVID-19. So it is used in Parkinson disease, useful in control of progression of disease and rheumatoid arthritis, right? So this is another. Bacopa moniera. I am only talking about antiviral activities. There are many other uses. So Bacopa monieri, which grows in the marshy places, all of you. So why? So therefore, identification of plants, knowing our biodiversity, plants and animals is of great significance so that we can utilize for benefit of human race. Bacopa monieri, leaf extract shows antimicrobial and antiviral properties. Leaves are taken against epilepsy, insanity, and nervous disorders. So I think then this is a Puraria tuberosa. Again, it is a common plant throughout Maharashtra, used as a tonic in rheumatic pains, in epilepsy, as an aphrodisiac tonic, galactic, diuretic, clears the voice, right? Centella asiatica. Again, this is another plant. Leaf decoction is given as an appetizer, usually in hair loss and graying of hairs. Growing up hairs, it is given as a cardiac tonic. Now, Wrightia tinctoria. Wrightia tinctoria, Kala Kuda, Pandra Kuda, we call it in Marathi, which is a very common plant throughout Maharashtra, is one of the best antidotes against the H1N1 virus in terms of inhibitory concentration. So this is plant. Gingiber gerumbet, the rhizomes, you know, of course, all of us, we know about haldi, its antiviral activity. Similarly, all gingiberous members have that antiviral activity, the antibacterial activity. The rhizome is used like the official ginger. It is given in cough, asthma, and rhizome is extremely applied to externally applied in itching. It is applied externally after leech bite. It is also used as a diuretic, right? Haldi. See this haldi, which are, if you come to Western Ghat in rainy season, in month of May end or June beginning, you will see this beautiful flowering of plants, right? Cissus quadrangularis, herpes simplex virus, feline immunodeficiency virus, coxsackie virus, influenza virus, influenza virus, respiratory syncytial virus. On that, this is used. Jasminum officinana. Jasminum species are source of sequoiarioidoides, glycosides which are used against hepatitis B virus. Jaundice, hepatitis B is very dangerous. So jasmine species are used. What I want to tell you that there are a number of plants which have antiviral properties, and if we can utilize our traditional knowledge, our research, whatever research have come, we can prevent entry of virus in our body, COVID-19. We can care for our virus. And one most important thing, lastly, I want to tell you to all of you is see why I said biodiversity is a real capital of human being. You can live without aeroplane, you can live without motorbike, you can live without a bungalow, you can live with many other things which are not essential. But you need food to feed your stomach, and all that food is synthesized by plants only. Plants are the only scientists who can synthesize food for you. So my stomach is my plants. Second point, my health. If my health is not good, everything else is useless. So all my medicines so far, since the origin of human beings, have come from plants. So my health is also my plants. And third thing, which I want to say, my breath is my plants. So breath, stomach, and health comes from plants. Fourth thing we need to remain happy is water. These are the things, basic things, which are the most important to enjoy everything on this planet. And therefore, biodiversity is of great significance for real progress, real happiness of human beings. I think with these words, I will stop here. Thank you very much. I am thankful to Vinod Varghese, for similarly principal of the college, for giving me opportunity to share my thoughts about COVID-19, Indian plant wealth, and antiviral plants which we can use in such a difficult time, so that we take care of health of our people, and that way see welfare of human being, avoid complications. Because in COVID-19, all of us, we know how many people have suffered. People did not get bed. People did, whatever medicines were given, they were, hurriedly given, which had a lot of side effects, and many people died because of that, not because of COVID-19. So all that we can avoid with these words. Once again, I thank you, all of you.
Thank you, sir. He was Dr. S.R. Yadav, a great taxonomist. Thank you, sir, for sharing your plants knowledge with reference to angiosperm diversity and antiviral species of plants to boost the immune system. Sir, with your lecture, we have acquainted with the botanical and the common names of the plant species around us. Once again, thank you, sir. Sir, we have got 3 questions. Thank you, sir. We have got 3 questions from the participants. So my first question is, why the quality of various biogeographic regions are destroyed in the present time?
Why the
quality of various biogeographic regions are destroyed in present time?
Number one, there are various reasons, but finally, only one reason is very important, that is anthropogenic activities throughout the world, not only in India. Because of increasing human population, we have great demands on biological resources in nature, and because of that, we have destructed a lot of biodiversity, we have destructed habitats, and that's why there is a continuous deterioration of our biodiversity and biological profiles. Second, climate change is also a result of human activities. And thirdly, our many activities are responsible for loss of biodiversity, especially our industrialization, pollution, cutting of trees, deforestation, and invasive weeds. all that today's extinction, mass extinction is because of anthropogenic activities, or we are responsible for that.
Thank you, sir. So the next question is that, can you tell me, during chickenpox infection, we use neem leaves. What is the mechanism present behind it?
See, I already told I am a taxonomist. I am not doctor. The person who has this question, he should go to some Ayurvedic doctor and ask him, because I don't know mechanism. Otherwise, I will read myself. I got the question, question. I will read, find out, and then tell. Just like that, I will not say anything, because that is not my field of studies.
Okay, sir. And the last question, in biodiversity, we mostly include all flora and fauna community, but what about microbes?
I say microbes also. You know, microbes are as important as other components of the. Without microbes, this, world cannot run. Unfortunately, they are microscopic and we don't study, but they are equally important. So it is a biodiversity. I have used the term biodiversity, which includes microbes. And one more request, my presentation can be given to every participant. There is no problem, right? Some student was asking, they can use my presentation. If you don't have, I will send it, and then you can share with students, participants.
Thank you, sir. We are very grateful for the time and efforts you took to share your thoughts and expressions for us, sir. Thank you very much.
It is my great pleasure. Thank you.
Thank you, sir. I feel privileged for having Dr. Ashish Kumar Jha, Assistant Professor, Department of Zoology, Hislop College, Nagpur, as our next speaker. Before that, may I request Professor Muneshwar to introduce Dr. Ashish Kumar Jha. Over to you, Professor Muneshwar.
Thank you, Dr. Tekade, madam. Respected principal, dignitaries on the online platform, faculty members, participants, and students, a very good afternoon to you all. It is my proud privilege to introduce Dr. Ashish Kumar Jha, Assistant Professor of Zoology in Hislop College, Nagpur. He pursued his master's degree in zoology and PhD degree from Rashtrasanta Tukroji Maharaj, Nagpur University, Nagpur. He has completed 3 minor projects. He has published over 24 research papers in national and international journals and over 9 research papers published in the conference proceedings. He has published 6 books for the degree students on topics like environmental biology, cell biology, entomology, animal physiology, etc. He has delivered invited lectures and chaired the session in many national and international conferences. Along with this, he has delivered guest lectures and in seminars arranged by various universities. He was awarded as Dedicated Researcher Award at International Conference organized by Shivaji Science College, Nagpur. With this brief introduction, I would like to request Dr. Ashish Jha sir to share his thoughts with us. Over to you, sir.
Can you check my video is switched off?
Yes, sir. Your video is visible, sir.
Is it visible? Because
yes, sir. Yeah, sir. Visible.
Okay. And my slides are visible?
Yeah, sir. Your slides also is here.
Okay.
Make it full screen.
Yeah.
Okay, sir.
Right. Right, sir. Thank you.
Okay. Welcome, sir.
Thank you, sir. First of all, I would take this opportunity to record my sincere gratitude to the management of Bharati Science College and notably the principal, honorable principal, Tatit sir, and members of organizing committee for providing me this opportunity to interact on a topic, immune system and its response to coronavirus. The importance of subject, how to say this importance of subject, I was just searching in the morning. This is the newspaper of the today's morning newspaper. I think that PM is also visiting today to the vaccination center. So one thing is very clear, because there is no registered medicine or vaccine against COVID-19, the immunity system is the best defense because it supports the body natural ability to defend against pathogens. So you can say that at present, as far as today is concerned, the immune system is the fundamental to this survival as it is protecting us from the pathogen. In coronavirus, in corona cases, it is the virus. Otherwise, it also protects us from bacteria and other parasites. One important thing that I want to tell you is that the immune system has evolved. This immune system has evolved to protect us from the pathogens because it is a powerful collection of defense mechanism that recognize and protect against potential invader that would otherwise take advantage of the rich sources of nutrition that our body or anyone, any host, whatever be the host, in the present case, it is the human, is that. So what this pathogen will take, if our immune system is unable to fight with them, then what this will do, this will take the advantage of the rich source of nutrition that we have. So naturally, this has to be evolved. So so moving for proceeding further, we will try to understand that what are the cells. There is some message coming. Why it is? So actually, lots of things are here, but the problem is that in the slides only we can see this. Okay, I should do this. Yeah. So moving to the part that is cells of immune system. Here you can see that when we are talking about the cell of the immune system, I want a pointer actually. Yeah, I want. So here we can see that in this diagram, there are cells of the immune system and some soluble mediators. So what we will do, we will first, as the slide is focusing, you can see that the principal component is the leukocytes. You can say that leukocytes are the central to all immune response. Other cell in the tissue participates, but the magnitude in which leukocyte participate is so high that you can say that it is the central, it is the chief thing that acts on immunity when we are talking about the immunity. First of all, we will try to understand the phagocytes, internalize antigen and pathogens and break them down. Now, to understand this, the most important long-lived phagocytic cells belong to the mononuclear phagocytes. So this you can see the first type of cell we are talking, it is a type of leukocytes, and these cells are all derived from bone marrow stem cells. That means when we are talking about mononuclear phagocytes, then these are the cells which are derived from bone marrow stem cells. And if you see their function, their main function is to engulf the infectious agent. What they do, they engulf the infectious agent and internalize it after engulfing, they will internalize it and they will later on kill it. So the first part is to identify this infectious agent has to be identified. So these mononuclear phagocytes have on their surface are receptors, and these receptors they identify the microbial peptides. They have the receptor on their surface, and this they identify this through the receptor. They identify the microbial peptides, and once it is identified, then they, by a process known as phagocytosis, they engulf it, they internalize it, and kill it. And the most important lineage of this is monocyte. In one of the leukocytes, you have heard that it is a lineage. One lineage is monocyte. Another example of is a polymorphic neutrophils. That is this neutrophil. This is a polymorphic neutrophil. This is also has a phagocytic nature. So what we have seen that one process, the first process, the cells that are involved in phagocytosis are basically mononuclear phagocytes. Among the lineage is the monocytes, and another we have seen is neutrophil. These two are basically acting as they phagocytes the or internalize the antigen and pathogens that that break down or which breaches our immune system. The second way is by B cells and T cells. B cells and T cells are responsible for specific recognition of antigen. Cytotoxic cells recognize and destroy other cells that have become infected. So talking about B cells and T cells, basically we are talking about adaptive immune response. Now this is mediated by lymphocytes. You can see that now we are talking about lymphocytes. Now, which includes two types. Basically, in lymphocytes, we will be focusing for this adaptive response. We will be focusing on B cells and T cells. B cells, you can see, as I told you, that immune system requires a cell as well as soluble mediator. So these B cells, when we are talking about the B cells, they are responsible for production of antibodies that act on extracellular pathogens. So B cells basically produce a soluble mediator, like example is antibodies. Similarly, we will be focusing on T cells. Now, this is concerned with cellular immune response to intracellular pathogen such as viruses. They also regulate the response of B cells. That means the T cells is also regulating the response of B cells and overall immune system. To understand, because when we are talking about coronavirus, so we are more focused on T cells. So I would like to include something more about T cells. Basically, if you see, there are several types of T cells, and they have variety of function. One group of T cells, they interact with a mononuclear phagocyte, that is macrophage, monocyte, or you can say, or you can also say polymorphic neutrophils. They act with the mononuclear phagocyte and help them to destroy, as we have already learned that by phagocytosis, by the process of phagocytosis, they destroy the intracellular pathogens. And these are called helper T cell type 1. These are basically type 1 helper T cells, also referred as Th1. The second type, you can see, the second type, they interact with B cells, and they interact with B cells and help them to divide, differentiate, and make antibody. These are type 2 helper cells, and they are known as Th2. So just you have to remember till the seminar that Th1, because we will again we will not go to this part. So you should remember that when I will be telling Th1, you should understand that I am talking about activation of
activation of mononucleophagocytes. But when I'm talking about TS2, I'm talking about activation of B cells. And once B cells, as we have seen previously in the previous slide, that B cells activate and they produce antibodies. So activation of B cell produces antibody. Now there is a third type of T cells. Basically, if you see, then their main function is cytotoxicity. That means they are responsible when they are responsible for when the cell is virally infected, then they are responsible for toxicity and killing that cell. So these cells, as the name is, this is a type 3, and they are known as cytotoxic T lymphocyte. The CTL stands for cytotoxic T lymphocytes. So these are the major type of T cells, and how they recognize. Then all the T cells have on their surface a specific receptor known as T cell antigen receptor, that is TCR. All the T cells will have this TCR on their surface. Now these T cells, as we have seen that B cells have a soluble mediator called antibody, the T cell has a soluble mediator. That means they release a soluble protein, that is cytokinin, that signals to other cell or direct the cell-cell interaction. So till now we have seen that phagocytic, the one way of cell acting as a immunity is by phagocytosis. Another is by B cell and T cell, where B cell is responding by making antibody, and T cells have either they activate the phagocytic cells, or they activate the B cells, or they can do the cytotoxic thing. So when we are talking about the cytotoxicity, this cytotoxicity is not only done by the T cell lymphocytes, but there are other like large granule lymphocytes and the eosinophil. The WBC has another, like neutrophil, there is another eosinophil that are or the acidophils. So large granulocytes and eosinophils, these are basically doing cytotoxication. Now, how they do, then all these cells type damage their different target by releasing the content. So you can see that they are granular, and these granules contain, they release their content. Basically the large granulocyte, lymphocyte, when we are talking about, then they release cytokines. And since they kill the cells, so it is commonly known as natural killer cell, NK cell. LGL is also known as NK cells because they kill the cells. And as I told you, another type is of eosinophil. These are a specialized group of leukocyte, but these are basically in the nematodes disease. Nematode disease like cystosomes. Cystosome is a type of trematode. In that you can see that cystosomosis, you can see the role of eosinophil, where it releases histamines and then does it destroy the cystosome. Now this was the third type. Now the fourth type of immune cell that we have is auxiliary. As the name suggests, these are auxiliary, they will be supporting. Now, what these do, basically they are of basophil mast cells are the common example of auxiliary cells. Now, what they do, they produce inflammation. Now, why to produce inflammation? Once the inflammation is done, then it attracts these leukocytes. If inflammation is there, then they will attract the leukocyte, and not only that, they will also attract the soluble mediator, that is antibody and cytokines. They will attract, they will draw their attention towards them, so that the inflammatory response is there. The third type is platelets. One of the auxiliary cells is platelets. Basically, platelets we have read in blood coagulation. The main role is in blood coagulation, but they are also released during the inflammation. That is, they are also inflammatory mediators because they are released. So, yeah. Shall I proceed? Am I audible? Am I audible? Am I audible?
Yes, sir. Yes, sir. You are very much audible. Yes, you are audible.
Thank you, sir. So this is so as we have talked about the immune cells of the immune system. Similarly, I told you that these cells during the immunity, these cells also have a soluble mediator. So we should also try to understand how this soluble mediator acts. So talking about the soluble mediators, this we have done the different functions. Now we will be going with the soluble mediators. Now, what are these soluble mediators? Basically, these are the molecules that are involved in the development of immune response. That means the molecules that are developed that are present in our serum for the immune response. In fact, whenever we will have any type of infection, then their number will be increased, and we call it as APP, that is acute phase of protein. So basically, soluble mediator increases its number whenever we have the infection. So one type is complement protein, which mediate phagocytosis. Basically, when we are talking about complement, then there are three pathways. These are alternative pathway, classical pathway, and MBL pathway. Through CA3 protein, basically the act that are present in our serum. But since complement plays a major role in bacterial infection, we are not focused here. So I'm skipping this part because we are dealing with coronavirus. So and this complement is basically for bacterial infection, where they go for phagocytosis. So I will be dealing with cytokines that signal between lymphocyte, phagocytes, and other cell body. We have already seen that cytokines are released by different group of cells. You can see here that cytokines are released by T cells, NK cells. LGL is basically NK cells. It is also other tissue and cells also includes the cytokines. So we will be focused more on this. Now, coming back to the cytokines. In general term, this is a general term for a large group of secreted molecules involved in signaling between cells during immune response. So these are the large group of molecules. And these cytokines are basically glycoproteins. The cytokines are basically glycoproteins, and the first type we will be dealing with interferons. Now, why we will be more focused on interferon? Because it has the ability to limit the spread of viral infection. Since its ability to limit the viral infection, we will be more focused on interferon. Now you can see here type 1 interferon. These are group 1, that is IFN alpha and IFN beta. These are type 1 interferon. Now they are produced by the infected cell. Whenever a cell get viral infected, it will produce IFN alpha and IFN beta. Later on, we will see how in coronavirus, how they are working. Now, another group is that is produced by the T cells. That is, and these two by helper T1 cells. That is IFN gamma. I had already told you that helper T cells, they will activate macrophage, that is mononuclear phagocytes. It will activate the mononuclear phagocytes, but it releases IFN gamma. So what we have seen that these interferons, whether it is a type 1 or type 2, type 1 is produced basically by viruses, and when we are talking about type 2, then T cells basically they activate mononuclear phagocytes to produce IFN gamma. And what they do basically, once interferon is released, they create an antiviral environment near the neighboring cells. So antiviral environment is created, and thus the cell and even the cell become viral resistant. They try to make them viral resistance. So this is all about interferons. So small introduction that I gave. Another is interleukin. Now, interleukins, these are also a large group of cytokines produced mainly by T cells. These are also produced by T cells. But when we are seeing its role, its main role is to divide and differentiate. So interleukins play an important role in division and differentiation of the cells. The third type is chemokines. Now, chemokines are large chemotactic cytokines, and their main role is in directing the movement of leukocytes. The leukocytes that we were talking here, the leukocytes that we were talking. So, what this the chemokines, what does this? When it is released, it directs the movement. They direct the movement towards the infected cells. So, this is the main role of chemokines. Then we have tumor necrosis factor. This is basically TNF alpha and TNF beta. I will be using now as TNF only. TNF alpha and beta have a variety of function, and inflammatory mediatory, when we were talking about, that is inflammation, they have an important role there, and also in the cytotoxic reaction. So when we will be dealing with coronavirus infection, we will be using this term, and these terms, as I have already told, that interferon, basically alpha and beta, they will be released by the infected cell, and type 2, that is gamma, will be released by the mononuclear phagocytotic cells, and they will have a virus resistant, they will try to do the resistance with the virulent. Interleukin will be doing the division and differentiations. Chemokines, they will be they will be informing, or they will be signaling the am I audible? Because the message is coming that I was muted. Am I audible?
Yes, sir. You are audible. You are clear.
After interferon, interleukins, chemokines, we have seen that they are playing an important role in signaling the leukocytes, and tumor necrosis factor, that is alpha and beta. They are basically mediating with inflammation and cytotoxic reaction. But there are other cytokines also, that is colony stimulating factors, CSF we call, like I told you, ILS. They also play an important role in differentiation of the cells, particularly leukocytes. So now I will be moving to the part that is the origin. Now, we will be just trying to understand that how this cell, human body and the immune system, how this cell of the innate immune system works. Now, as we have seen that there are the infectious agent can be virus, the infectious agent can be bacteria, the infectious agent can be parasites or pathogens like protozoans, nematodes. So there are various type of this. So there is a need of, you can say that there is a great heterogeneity of the cells. There is a heterogenesis. That means, as you know, the virus has a very different, it has only protein in its surface. Bacteria has a different, this gram positive and gram negative. So they have a heterogeneity in the cells of the human system. And most of the cells will be hematopoietic, will be originating from hematopoietic stem cells, that is bone marrow or the fetal liver. So they will be originating from it. Why this heterogeneity occurs, or why this, as I have already told you, the main reason behind the heterogeneity is, you can say, the functions that they are going through. For example, when we are talking about mononuclear phagocytosis cell, the function is phagocytosis, or there will be a function called antigen presenting cells. Now we will be dealing with that, APC, or there can be a function of lysis of virus. So it's there can be a function of lysis, or there can be a function of secretion of antibody. So according to the function, there is an heterogeneity among the cells of immune system. Now we will be moving to the part that is, you can see here, this is basically hematopoietic stem cells that can be bone marrow or fetal cells. Here we can see that the megakaryocytes, that is platelets, once they originate from bone marrow, then they move into the blood circulation. So they are found in the blood circulation. But in case of, you can see polymorphonuclear granulocytes as well as monocytes, they are not only, they first go to the circulation, but from the circulation they passes to the tissue. So they remain to the tissue, they are released into the circulation, and then from the circulation they passes into the tissue. Now you can also see that mast cells. Now mast cells are identified, they can be identified in all the cells. And mast cell is very important because they produce histamine as an allergen. So they can be seen in all the cells. Next, you can see the B cells. Now, these B cells, they mature in either they mature in fetal liver in the case of fetus, or after postnatally they mature in the bone marrow, and then they go to the blood circulation. And whereas when we are talking about T cells, they mature in the thymus. So T cell mature in thymus. Now, whatever be this, these lymphocytes, that is T cells and the B cells, they recirculate through secondary lymphocyte tissue. They will circulate through secondary lymphocyte tissue. Now, we have introduced a new word, secondary lymphocyte tissue. I will be talking about it later on. So just understand that lymphocyte is released into the secondary lymphocyte tissue. I just forgot one cell, that is NK cell. It is released in the blood too. So from blood, it goes to the tissue, natural killer cell. Now, as I told you that this lymphocyte recirculate through secondary lymphocyte, and you can see that here is interdigiting cells, dendritic cells, and macrophage. They are known as antigen presenting cells. So we are now introduced to a new word, that is antigen presenting cells. So now we will try to understand first what is antigen presenting cells, and then we will be dealing with secondary lymphoid tissue. Now, talking about antigen presenting cells, so these are basically, this link the innate. You can see here that it is linking the innate immunity and adaptive immunity. Now, how its role is played, you can see here that when a microbe, basically you can see the virus, when a virus infection is there, then these antigen presenting cells, through its surface receptor, they recognize it, they process this antigen, and by processing this antigen, it makes it recognizable by the T cells. So this comes under adaptive immunity. That means antigen presenting cells, they receives the antigen, and then they process it so that it can be recognized by the T cells. And the other role in it, as I told you, that it links it to the innate immunity. In innate immunity, these antigen presenting cells, they release cytokines. These cytokines later on activate the mononuclear phagocytes. They can also activate this NK cells. And these T cells, I had already told you that the T cells have the ability that they release the cytokines to activate the B cells. So what we see, that APC, that is the antigen presenting cells, their main role is to link the innate and adaptive immune system. Now, as I told you that we will be talking about this secondary lymphoid tissue, we have just seen that thymus and bone marrow are the primary lymphoid organ, where because they are the hemopoietic tissue, bone marrow is the hemopoietic tissue, and in thymus we have seen that T cells, the T cells gets mature there. So these are the primary lymphoid organ. Beside this, we can see that lymph nodes and spleen. Now, talking about this lymph node and spleen are the secondary lymphoid organs. Now, whenever there is an antigen in blood, so there comes the role of spleen. Lymph node plays role when there is an antigen in the lymph. There are other secondary, like you can see here that bronchus associated, let me make it here. You can see here that Pierre patches, lymphoid nodules, bronchus associated lymphoid tissue, MALT is another one, and Walder's rings is another. So how these act, as we have seen that spleen and lymph act when there is an antigen in blood and lymph, then is the role of spleen and lymph node come. Here, when there is antigen present in the mucus, whenever there will be an antigen that is present in the mucus, then its role comes. So you can say that surface, when antigen are present in the surface mucosal barrier, then the role comes. So this is about basically we have till now we have understand the cells that are involved in immunity. We talked about the soluble mediator. We talked about the lymphoid organ, that is the tissue and the tissue, secondary lymphoid organs. We talked primary lymphoid organ. We talked. Now we will be dealing with the coronaviruses. Now, talking about coronaviruses, as you can see here, the coronaviruses basically the name comes from its crown-like spike on their surface. It is in Latin, corona means crown. So it is the surface, that is the crown-like surface. It is a coronavirus. They belong to a family that is Coronavidae. Now, this Coronavidae, all the family members of the Coronavidae, they possess extraordinary large single-stranded RNA, that the genome that ranges from 26 to 32 kilobase in length. So when we are talking about the family Coronavidae, they possess basically a large single-stranded RNA that ranges from 26 to 32 kilobase in length, and they are causing infection not only in human but also in other animals also. So currently, when we are talking about the human coronavirus, so currently you can see that when we are talking about human coronavirus, their number you can see is basically 7. They are 7 in numbers, of which HKU1, human coronavirus, OC43, 229E, and NL63, they are usually causing mild respiratory illness. We will talk about it in the next slide. What is this mild respiratory illness? There is another group that is MERS virus, that is Middle East Respiratory Syndrome. Now, this affects not only the upper inflammation, but it causes the middle inflammation also. We will, again, as I told you, we will be talking about it. Now, I am focused on this SARS coronavirus. Talking about SARS coronavirus, this is the virus that which causes SARS stand for severe acute respiratory syndrome. It was detected in Guangdong province of China in November 2002, subsequently spread to more than 30 countries. So in 2002, there was an outbreak of SARS-CoV and more than 8,000 cases were reported worldwide, and it caused almost 10%, that means 800 deaths. It caused about 800 deaths. Now, SARS-CoV is a zoonotic disease. That means its origin is from horseshoe bat, and is a natural reservoir. And the immediate source of SARS-CoV was the civet cats that were, again, as in this case, this was also sold in a animal market, civet cats, from where this caught. Now, the main infection or the main damage it did was pneumonia. The upper respiratory tract was basically damaged, the cell peeling, cilia damage, formation of multinucleated giant cells. Then aplysia was another, that is the squamous cell aplysia was there, fibrotic lung disease were there. So this is all about SARS-CoV, but today, now the main problem is SARS-CoV-2. This virus emerged in December 2019 in Wuhan of China, and was identified as a novel beta coronavirus, as you can see here. First, it was called as 2019 nCoV, but when the genomics of 2019 nCoV was sequenced, it shared 80% almost. It said almost 80% of the genetic sequence is SARS-CoV, and International Committee of Taxonomy of Viruses renamed it as SARS-CoV-2. So now it is known as SARS-CoV-2, that is COVID-19. The COVID-19 infection is the infection name, and the virus name is SARS-CoV-2. As I told you that we have to first understand how this illness is caused. Now, talking about the first 4 human coronavirus, now talking about the first 4, that is SCOV-229, OC43, NL63, and HKE1, you can see that they are causing the illness in the upper respiratory tract only. So this is a mild cold symptoms. They cause the mild cold symptoms, and they replicate only in the nasopharynx region. So this is not a that seriousness. And when we are talking about MERS-CoV and SARS-CoV, that is one, SARS-CoV was originating in 2002 and this in 2014, you can see that they causes lower respiratory tract disease. They were causing the lower respiratory tract disease, but when we are talking about SARS-CoV-2, you can see that they are causing both upper and lower. The most important, the main problem is the asymptomatic to severe anemia. Replication, you can see that it is throughout the respiratory tract, and the immunity duration is still, we are not very sure about its immunity duration. Now, again, I would like to go for this, the time, just I would shorten this, I would not read everything about it. As I told you that this was, the virus emerged in 2019 in Wuhan of China. So the Wuhan Health Locality Authority issued an epidemiological alert on 31st December. They gave that alert. Now on 9th January, this is important because on 9th January 2020, Chinese researchers have shared the full genetic sequence of coronavirus. And after that, on 30th January, you can see here that 30th January, WHO declared it as global public health emergency. And if you consider till now, then till now it has, the deaths are in India only, we have seen that the deaths are so high. So moving forward to this, I would like to say that how this SARS-CoV, I am more focused on how this SARS looks like. So whether it is the SARS-CoV of 2000 or SARS-CoV-2 of 2002, they have a same structure protein. That is, they have spike. You can see this. S is for spike glycoprotein. M is for membrane protein. E is for envelope protein, and N is for nucleocapsid protein. The S protein, when we are talking, is a trimeric form of virus. It has a 2N, that is, and as we all know that the protein has a amino end and a carboxyl end. The N end of the S1, this is a trimeric protein. They have S1, S2, and S3. The N end of the S1 contains the motif that is responsible for binding. So it binds to the host, that is to the respiratory epithelial cell, where we will be looking at it through this S1. And S2, when it binds, then there is a subdomain S2, which contains a heptad repeats and coil structure, and is important for membrane fusion. It is important for membrane fusion. The M, now talking about the M protein, is a triple spanning membrane protein and a key role in coronavirus assembly. They play an important role in assembling of coronavirus. The E protein is required to bind the sugar motifs of the cell membrane. And coronavirus N protein is required for coronavirus RNA synthesis. So we have this is required for RNA synthesis and has RNA chaperone activity and may be involved in template switch. Talking about the difference you can see with other coronavirus is this hemagglutinin acetylase glycoprotein. This is not reported in SARS, either CoV or CoV1 or CoV2. It is not reported. But In all other 5 coronavirus, we can find hemoglobin, acetylase, glycoprotein, but this is not present in the present coronavirus. Now, how the infection goes? Then, since SARS-CoV and CoV-2 are so similar, so the scientists have given the hypothesis that is the biochemical interaction and pathogens are like similar. Now you can say that the you can see here the binding of SARS-CoV triggers the renin-angiostensin system. It imbalance. Now, first I will try to make you understand what is this renin-angiostensin system. It imbalance to ACE2 contribute to pathogenesis of COVID-19. Angiostensin converting enzyme 2, that is the ACE2. This is angiostensin converting enzyme. Is composed of wide range of peptides. You can see here, ATR1, ATR2. This there is a, you can say that angiostensin 2 and angiostensin are the major effector of 2 main RAS pathway. This is a RAS pathway, which we will be proposing. Now it has a 2 pathway. One is a classical axis pathway where ACE and angiostensin are involved, and another is an alternate axis pathway. In present coronavirus case, that is the SARS-CoV-2, we are using alternate axis, that is ACE2 and MASR receptor, that is MAS receptor pathway. So we are using this. So what happens here, that the ACE2 receptor, that is the type 2, this type 2 receptors, even you can say that where it is present, basically it is present in the epithelial cells of the lungs. It is present in the epithelial cells of the lung, and as I told you that it triggers a renin-angiostensin system. So first what will happen, first this binds to the ACE2. Once the coronavirus, that is the SARS-CoV-2, binds to the ACE2, it causes a cascade of inflammation in lower respiratory tract. As we have seen that it not only affects the upper respiratory tract, but it also affects the lower respiratory tract. So cascade of inflammation is there in the lower respiratory tract, and the complex binding of ACE2, then there is a proteolytic processed by this TMPRSS2. Now this is proteolytic processed by transmembrane protease, that is TMPRSS2, leading to the cleavage of ACE2. So it will cleave the ACE2 and activation of the spike protein, and thus it facilitates the entry into the target cell, that is lungs and tissue. So it is facilitating its entry to the target cell, which requires ACE2 and TMPRSS. Am I audible? Am I audible?
Yes, sir. Now you are audible, but sometimes your voice is cut and your voice is not clear.
Might be of network issues. So we were talking about this, how coronavirus enters the lung injury. We have seen that ACE2 recognizes the spike protein, and then there are TMPRSS2, which cleaves it, and then there is an this is how they go through the lung injury. Now we will move to the slide that basically now that immunity against virus in COVID-19 patients, we will just now see that how our body is responding. Now you can say that the early stage of viral infection is often a race. It is a race between the virus and our host defense system, in which the virus tries to overcome the host in order to establish the infection and then spread to other tissues. So it is a race where our body will try to resist the viral infection. When the coronavirus initial defense, our body will be trying to resist, and the virus will try to overcome. our defense system. So there is a race between them. The main motive of virus, that is the SARS-CoV-2 virus, the main motive of this virus is to establish an infection first. Once it is able to establish the infection, then another role will be to spread to other tissue. Then the role will come to spread to other tissue. Now, how our body will stop it? So the initial defense against virus invasion is the integrity of our body surface. How our body surface is integrated, it is the initial defense. So this can there are anatomical barriers that our body provides. That can be acid pH. Our body will be providing with acid pH so that the virus can't breach the immune system. They will provide a proteolytic enzymes. They are providing bile. They are providing mucus. So these are the first level of immunity that our body provides it. But once that is breached, that means then what our body will do, it will once infection triggers the activation, it will activate the DC cells. That is the dendritic cells. Once the primary, as I told you that whether it is the acid pH, proteolytic enzyme or mucus layer, once it overcomes it is breached, then our body activates the dendritic cells. Now, these dendritic cells, not only dendritic cells, there can be macrophage cells also. They activate it. Now they release the cytokines. What they will do? They will produce a variety of cytokines, chemokines, antiviral peptides. So they will do this to come over it. Now here comes the main role. So which is the most important role? I have already told you that it is the interferon. So key innate antiviral effectors are the interferon. So we will try to understand that how this key innate antiviral effector, interferon, also tumor necrosis factor, they play their role. So here we can see that interferon 1 production typically starts to be induced within the first few hours of viral infection. So this is the viral infected cells. When it gets infected by the coronavirus, then what will happen? This within the few hours, you will see that they will produce the interferon. That is the type 1 interferon. Now, how it does? There will be also a second response. There is a specialized plasmatocyte DC cells. That is a dendritic cells, which is also triggered to produce IFN alpha B. Now, first we will be taking the case of the infected cells. These infected cells have RIG1, that is the retinic acid inducing gene 1. These are the receptors. Once the viral protein comes, they receive it. They receive the viral nucleic acid, and once they received it, they trigger IRF3 and NFKB. And now, when this is triggered, they tell the nucleus to translocate mRNA. Once translocation is done in INFA, that is interferon alpha and beta, that is the type 1 is produced. Not only this, they also produce inflammatory cytokines. This is all done by the viral infected cells. Similarly, there is, as I told you, not only this, but the plasmatocytes, dendritic cells, they have toll-like family receptor that has TLR. Now, this TLR 7 and 9, you can see here, they do the same thing. They will activate, there will be translocation of mRNA, and again we can see that IFN, that is the type 1 interferon, is produced. And beside this, inflammatory and cytokines are also produced. Now, this is the first step they do. Then how this type 1 act? Now, the first part of the type 1 will be that it will directly, this is the infected cell, it will directly act on the infected cell and try to inhibit the viral infection, virus replication. That is, it will try to inhibit the virus infection in the infected cell. Not only that, this type 1, it will also trigger the NK cell, that is the natural killer cells. It will trigger the phagocytic cell, that is the macrophage. It will trigger the CD4, CD8 cells. It will, now this is major histone complex, MHC. So when we are talking about CD4T and CD8, we are talking about MHC1, CD8 and MHC2. This is CD4. Not only this, they also trigger B cells to produce the antibody. So, and also it act on the dendritic cells to stimulate and increase the expression of MHC1 and MHC2. So what we have seen that once there is a viral infection, this infected cell and the plasmatide dendritic cells all have the main motive is to produce type 1 interferon. This type 1 interferon, they activate macrophage NK cells. Even they inhibit those who are infected by virus, they inhibit the viral replication there. They also produce major histone complex 1 and 8. They also produce activate the B cells. Now, why do they do this? Once there is a viral infection, they are creating an antiviral environment. What we see that type 1 creates an antiviral environment by this activity. Now, as I told you, they are activating the NK cells. Now, what is the role of these NK cells? They are cytotoxic, as I have told you earlier only, that they are like a cytotoxic T lymphocyte. These NK cells are also cytotoxic for viral infected cells. Now, NK cells act to combine virus replication directly by recognizing and killing the infected cells. They recognize it and they will kill that. They also produce cytokines, and basically this cytokine, that is INF gamma, that is the third type of interferon, IFN gamma, and tumor necrosis factor. They produce tumor necrosis factor also. Now, the main purpose is of this is that it acts as an immunomodulatory effect. So first they kill the viral infected cell. They also combat the viral replication. They produce cytokines like interferon gamma, that is the type 3 tumor necrosis factor, which act as an immunomodulatory effect, stimulating the activation of macrophage. So what we see later on, again, I will show you graphically, we can see this, that this is how the NK cells act. They activate the type 3 interferon, type 2 interferon, that is IFN gamma. They also act on this will act on the infected cell and it will kill the infected cells. So the main purpose will be to kill them. Beside this, they will also mediate immunomodulatory effects. They do that and stimulate the activation of macrophage and DC cells. So this is the main role of the NK cells. Now the macrophage, when the type 1 interferon activate the macrophage, this macrophage have 3 levels to destroy the virus. First is by phagocytosis. They kill or they phagocyte the virus, and basically they phagocyte the virus infected cells. They also kill the infected cells. So what we see that first is that they phagocyte the virus. Second is that they kill the virus infected cell, and the third is production of antiviral molecule. That is, they are again producing the tumor TNF alpha and the INF, that is type 1, that is IFN alpha. So they are again producing it. So this is the role of macrophage. Now we will also see that what this type 1 has done. The type 1 has activated the B cells. Now, how this adaptive B cells and major histone complex, that is the T cells, these are activated, that means we are talking about adaptive immune response. So talking about adaptive immune response typically begins with few days after innate response. Once this innate response has happened, then it will take some time. It will take few days to appear after 4 days of infection. Am I audible? Am I audible? Excuse me, am I audible?
Yes, sir.
Okay. Okay.
Yes, yes, audible.
Now we were talking about the adaptive immune response. Basically, T cell will start forming after the 4 days of infection. So first we will be talking about the taking about the B cells, how these B cells work, because they produce the antibody. Now there are two ways that it can function. One is that antibodies and complement can limit viral. I told you that complements basically these are for bacterial pathway, but they play an important role. Now antibody alone without complement also, antibody act on the free virus. They block the binding to the cell, they block entry of it, they block uncoating of viruses. Similarly, this can also act together with complement, and they damage to virus envelope, block the virus receptors. Another way of their this antibody is that they act on virus infected cells. The first was on the virus. The second is that they act on the virus infected cells. So antibody together with the complement damage the infected cell, opsonize them, the infected cell, and later they go for phagocytosis. But the most important is that antibody, this virus infected cell, antibody bound to infected cell, and antibody dependent cell mediated virus inhibition by natural killer cell. So here we see the role of natural killer cell, macrophages and neutrophils, where they act on it and they kill the viral infected cell. But the most important role in the adaptive immunity is by the major histone complex 1 and 2. Now talking about the 1, that is the CD8 plus T cells that target the virus infected cells. Now how they do, you can see here that this is the CD8. Now how they will do, now they kill the infected cell through release of perforin, granzymes and cytotoxic protein. So how they kill? They kill it by the release of perforin, granzyme and other cytotoxic protein. Now they trigger the death of infected cell through binding of soluble factors like TNF alpha now and interferon gamma. Now what they do, this will trigger the death by TNF alpha, tumor necrosis factor is activated so that the viral infected cells can be killed. So this is by TNF alpha. The third is by producing the soluble factors like IFN, interferon, basically interferon gamma, that cure the infection. Now what they will do, now they try to, they go inside this interferon gamma and they try to stop the viral replication, and thus they will try to cure the infected cell. So either by killing the infected cell or by curing. So this is the role played by the CD8-4 T cells. Another is CD4. CD4, this is the major histone complex 2. These are the major effector cell population in response to virus infection. These have been identified as major effector of the cell population in immune response. They produce cytokines, IFN gamma. You can see here that they are producing the cytokines, interferon gamma. This is the third type 2 interferon. And also TFN alpha. They also produce TNF alpha, which mediate direct antiviral effects and also to activate macrophage. They activate the macrophage And once this macrophage is activated, they will undergo the phagocytosis. So this is the way that CD4 T cells act. So either by type 1 interferon, they will produce the type 1 interferon, or they will activate the macrophage. In return, macrophage will, as we have seen, we have already told the role of macrophage. The macrophage, you can see, we have already told that phagocytosis of virus, or the kill the infected cell, or production of antiviral. That is that type 2 interferon they will do. Now, this is how our body was responding to the viral infection. So now the question comes, what is the strategy that this coronavirus has? Now the question comes, what is the strategy that this coronavirus have evolved so that they can escape our immune response? So talking about their strategy, the SARS coronavirus has evolved. That is the first strategy that they have evolved is that they block the type 1 interferon production. So the first part, the first thing that they do is that by blocking the type 1 interferon. Second is that they impair the recruitment of plasmatoid dendritic cells. So we have seen the role of dendritic cells in production of interferon. So if they are not able to, in the first case, we have seen that they will block the interferon production. How? Then they will impair the recruitment of plasmatoid DC cells. We have seen that DC cells were producing the type 1 interferon. Or they will reduce the circulating plasmatoid dendritic cells. Or they will interfere with the antigen presenting cells. They interfere with the antigen presenting cells. They are producing the cytokines homologue. They can produce the cytokine homologue. So this is the first way that how virus tries. The second strategy of virus is by avoiding the recognition by host immune defense. If it is not able to block this or impair our immune response, then they try to avoid this immune response. How? By latency. Now, during the latent infection, within certain host cells, during which there is a little or no production of virus, latency infected thus essentially invisible to the host immune system. So because of latency, it is invisible to the home immune system. So we are finding so many asymptomatic patients because of this latency. It is invisible to the host immune system, and so they survive. Another way is that infection of immune privileged site. That means there are certain sites, immune defense is, they get access to those sites, and where they may also be an immune suppressive environment. So they get access to this immune privileged site, and there they grow. So our immune system is unable to recognize them. The last method that this virus is using is down-regulating the MSC class 1. We have seen the role of CD8+. Now what they do, they down-regulate the MSC class 1. They reduce the generation of epitope polypeptide in the cytoplasm. They block the peptide uptake of endoplasmic reticulum. They prevent maturation and assembly and migration of major histone complex. Lastly, it has been seen that recycling of major histone complex 1 molecules from the cell surface. So these are the three methods this virus has evolved. The coronavirus has evolved. First is that they impair the host immune response. Another is avoidance of recognition. And the third is virus avoid recognition by T cell reducing infected cells. So because of this, it is the vaccination process is time taking. Now we will talk about how this vaccine that we are talking about, the coronavirus. So how this vaccination is being developed. We will try to understand this part. Now talking about the vaccine process, basically we are taking this spike protein first, and then we are making the mRNA sequence of this spike protein. So at present, scientists scientists are able to generate the mRNA sequence of this virus spike protein. After that, we use liposome. Liposomes are lipids bubble in which they put the virus, the RNA, mRNA they are putting there. So the RNA sequence, a blueprint from the making the spike is put into the lipid coating for delivery. And then once it arrives the cells, once it will arrive, the cells read the information of the mRNA sequence to produce millions of copies of the spike protein. So these are they produce the spike protein, but you see that there is no viral DNA. Viral RNA is not there. It is only the spike protein. And this spike protein is recognized by the cells and they produce the antibody. So this is the way, this is the main principle that is working on. We are at present, we are working on. Most of the lab have reached the third phase of this, but some are in second also, some are in second, but most of them have reached the third trial, like third clinical trial. Now, there are certain guidelines by the WHO because still vaccine is not there. So the WHO has given certain guidelines. First is that our vitamin and minerals are vital for to fight against this infection. So vitamin B, which protects us from COVID infection. Vitamin C protects us from flu-like symptoms. Vitamin D and E, this is also playing an important role to fight against coronavirus infection. Our iron intake, WHO has that the iron intake should be accessed so that risk of having COVID-19 is reduced. Zinc is necessary for maintaining the immune system. So therefore, we can say that regular meal should be the meal that we are taking, it should be with vegetable, legumes, nuts, whole grain foods, because we require all this. You have seen B, C, D, E, all this we require. Malnutrition is a dangerous for patient of COVID-19, and thus proper nutrition should be provided in this case. We should avoid the fizzy drinks, that Coca-Cola, this type of things we should be avoiding. Unsaturated fats, white meat and fish should be consumed more and more to fight, to make our immune system strong. Saturated fat, red meat, more than 5 grams salt per day, and industry processed food should be avoided. Individuals should be active. Exercise is one of the things, because we all are staying home. Our children are staying home, so we should perform active and perform physical exercise to boost the immune system. And we should not be stressed by this COVID-19. We should have a proper sleep. Although there is no registered medicine for COVID-19 treatment, but we have seen that hydroxychloroquine remdesivir was prescribed earlier. But now, presently, if you will see, then presently we see that this has been again kept under WHO restrictions. So what are the potential challenges in immune system development? We have seen that an effective immune system must have the ability to interpret the changes that the world is facing, today's world we are facing. And then this immune system should respond properly. It must overcome to work in different environment with different pathogen, because suddenly what we have seen that COVID-19, there is SARS-CoV-2 came and things changed. So our immune system must work to different environment with different pathogen. Most of the time, the immune system encounters something new and consider it harmless. But in some cases, that response can be dangerous. An efficient immune system must have the ability to distinguish this. It should have the ability to adapt the strategy to the strange environmental change to fight against this infection, we can say. A healthy immune system has symbiotic microbial forms and react to harmful infection. When pathogens enter the body, they attempt to use it as a host and the immune system and many threats. A different door is used for every system. So we are waiting for the vaccine. So this is all about that. I want to tell you, I can conclude it that the conclusion is just like that. It is review on boosting the immune system. So a potential resource of treatment of patient with COVID-19. The process and mechanism of the immune system can be a good source of knowledge for immune system development. So that's why I linked with every aspect of immune, right from the basic to the advanced. We have gone through it. So, and lastly, I can say that till vaccination is not there, finally nutrition is to boost our immune system. So we should take proper nutrition. This is all. We should try to boost our immunity. So this is all about it. Lastly, I would like to acknowledge my thanks to the organizing committee, to the principal, and of course to the management for providing me the opportunity to speak on this. So if you have any query, you can ask me.
A very big thank you to Dr. Jha sir. Very nice and informative talk on immune system, cells of immune system, and how we can protect our body from the coronavirus. Once again, thank you, sir.
Thank you.
Friends, thank you, sir. So friends, let's have the pleasure of listening to our last guest speaker. He is none other than Dr. DVNS Suresh, Professor, Department of Zoology, Dr. Ambedkar College, Nagpur. It's a great pleasure to have you, sir. Before that, let me request Dr. PJ Aute, Head Department of Zoology, to introduce Dr. Suresh, sir. Over to you, Dr. Aute.
Thank you very much. Sir, Jha sir, may I request you to please stop your screen. Thank you very much, Dr. Rupali ma'am. Good afternoon. I extend warm welcome to all. I find immense pleasure in introducing our guest, Dr. DVNS Suresh sir, who has been working as professor at Dr. Ambedkar College, Nagpur. Dr. Suresh has worked on fish molecular reproduction, fish neuroendocrinology, and received training on molecular genetics and cytogenetics. He has completed his PhD from Department of Zoology, RTMNU, Nagpur. Sir has a research project funded by UGC on topic Trust Network, morphometric studies of diverse fishes in water bodies around Nagpur. Dr. Suresh has published lots of research papers in national and international reputed journals. He has published 3 books of zoology which are awaited. He also worked as postdoctoral fellow at ICMR, New Delhi. Now I request Dr. DVNS Suresh sir to start his presentation. Over to you, sir.
Thank you very much, sir. Am I audible?
Yeah, yes.
Yeah, thank you very much, sir. First of all, I would like to congratulate the organizing committee for settling this national seminar on COVID-19 and the impact on life sciences. So, I would like to extend my thanks and thanks to the organizing committee, the management, and the principal sir and Varghese sir who personally spoke to me. Thank you very much, sir. Let us begin with. Can I share my screen? Is it shared? Hello.
Yes, sir. It is.
Yeah. Thank you, sir. So, we have seen in Yadav sir's presentation that he has told us about so much of plant wealth available for combating these hardships and the scenario that we are going through the pandemic of coronavirus. So we have seen he has been very good enough to introduce us all to the different types of different diversity of different plants and the extracts and different flowers and different ways of treating a plant, all those which are useful for combating the present scenario of COVID. And later, Dr. Jha has introduced the intricacies of the immune system, the complex system which deals with the real combating inside the living body. And my focus is would be for a layman understanding of what COVID is and how the coronavirus, the virus which gets into our body, and how does it replicates and how does it affect us and affect our system, affect our health, and how does the immune system fall back to combat? What are the things which are needed to be as a precautionary measure, and a little bit of the origin of the disease, the origin of this infectious disease, and the people behind them. The bit is a bit that snippet is a bit controversial, but definitely we would touch up and we'll go. So let us begin the COVID. We'll try to understand in a very layman term, not in a very technical or a biological way. Anyways, so it's a different perspective to understand this COVID. So before that, I would like to present a small clip in which you would be visually you will be aided to know how exactly the virus gets into the living cell and comes out with a multiple copies. So I will not be using the the sound of the clip because I'll be stopping the clip in between and speaking. So and we consider him as he is infected. And first of all, I would like to tell this video or this movie clip is not made on the basis of the COVID virus, but this is made on the basis of the viruses. How do they infect and how do they propagate in the living system? So this is a person. If we consider this person as infected with some virus, consider it as a coronavirus, then it can be transported to the other people via bubbles, the droplets in the air, which might accidentally be inhaled inside of healthy human, and where on the nasal region or the upper respiratory tract region where the epithelial cells are there, and those cells are the first one to face the impact of these viruses. They usually it's been established the viruses usually get into the living cell with the help of some reception. there should be a reception in which these viruses are usually taken into. The taken inside a cell is called internalization. So internalization can occur when there is a reception between the cells, the living cells, and the virus or the foreign bodies which are coming into. And Dr. Jha has already mentioned about the molecular level of how the reception can be like justified to get the viruses into the living system. So we'll go further. So now in this clip, usually the yellow parts on this is a virus, and this is our surface of our living cell, the internal epithelial cells of nasal pharynx region. So these yellow things, as we have already heard about them, these are the spike proteins. These are spiky structures on the virus surface, which has got an envelope. It has got envelope which is proteinaceous, and membranous proteins also there. And then there are some spiky proteinaceous structures which is coming out of those viruses. These are called spike proteins. Sometimes they're called spike proteins, and sometimes they're called S proteins. Whatever name you call it, because for understanding, we need to know what exactly they do. So these receptors, these structures on the surface of the living system of us are epithelial cells. These are the receptors which recognize these spike proteins as self. Why that we would see in further clips, further slides. So what happens as they are recognized, and the virus is considered as self? Maybe they are considered as something which is coming inside the cell, which is helpful for the maintaining the mechanism of the living system. So the viruses which have got those proteins have got the similarity or the resemblance with the receptors that are there on the living system of the epithelial cells. So that way they have been received inside and they have been internalized. And then, it's another understanding like how these vesicles, which they are internalized or phagocytized, and these the vesicles are usually drawn to the center of the cell, that is nucleus, where we have got a big system of making copies of the nucleic acids. So that way these are taken down to the center of the cell, that is nucleus. And the nucleus is protected by its own membrane. That's a double-layered membrane, but it has got pores, which allows the nucleic material some size or some particular specific. It's very specific. It's permeable to only some specific sizes and shapes. So those nucleic materials are as these viruses, the retroviruses, because they have RNA as their nucleic content. So these nucleic content has been with the small proteins associated with them are taken inside the nucleus, and there in the nucleus, that means for a layman understanding, we have every cell everywhere. So at the epithelial cells of our throat region, there the cells have the receptors by which these viruses are recognized as self, and they have been taken inside. And the RNA which is there in these viruses, those nucleic acids, those RNAs, the RNA and the associated proteins, they usually get inside the nucleus where the system of making copies of nucleic acids exist, because even we need that system. But the viruses do not have that system in themselves. So they always need some other cell. Because they do not have that system within themselves, they are like considered as non-living. But when they have the system which is needed to replicate, and they need a host or a living cell in which they can exploit that living cell, and they can avail the facility of making the copies of those nucleic acids which they carry the information for their existence, for their protein synthesis, and for everything for their living. So that way those copies are made. The genetic material of the virus have been taken into the nucleus, where with the help of our own machinery, sometimes they carry some molecules which are helpful in their copying machinery. So that way they make copies, and they, these structures are ribosomes. Ribosomes, smaller and the larger portions of ribosomes, and these are the mRNAs which are coming out of the nuclear pore and getting associated with the ribosomes and getting translated into protein molecules. The proteins which are usually coded in the RNA of that virus, the proteins which are required by them for making it's the proteins which are associated with the RNA, and the proteins which are helpful for making the envelope of that virus. So that way, these protein molecules and the RNA, these are the associated proteins with the RNA. They assemble them, they organize themselves, they again now come out of the cell. Now they're ready to come out of the cell, but with the help of the translation, the synthesis, the protein synthesis of molecules, with the help of the RNAs which were copied inside the DNA, those protein molecules sometimes need some modifications. So they are usually synthesized on the surface of endoplasmic reticulum, which is near to endoplasmic reticulum, a structure which is near to the nucleus. And this is the Golgi apparatus, Golgi complex, where they get, if at all they need some modified proteins. Why they need some modified proteins? Because they need to have the spike proteins which resemble the receptors which are there on the living cells of our epithelium. So that way they need it. So that kind of modification are again done in the Golgi complex, and then they have been like passed on to the surface to get them out of that living cell. Now this is, see how they emerge out of the cell. The one virus that has entered inside this living cell, epithelial cell, now when they're coming out with multiple copies inside the RNA, inside the cell, with multiple copies of RNA and protein molecules to organize them, they come up in multiple forms. See, multiple copies of, you can call them the baby viruses. These are the baby viruses which are coming out of that cell which has been infected earlier. So now those infected viruses again come, and they can infect the other epithelial cells to get more and more in number. But as in the earlier presentation, you must have tried understanding that how those can be blocked, how that innate immunity helps us in dealing with that infections at the early stage. But when the infection or the virus surpasses that particular innate immunity responses, now the adaptive response of immunity is required. So in the earlier presentation, it has been clearly shown, or it has been clearly made understood how the phagocytosis has been done of these viruses. And this is the macrophages, which usually, these are the antibodies which recognize them, and the macrophages are
micro
all around. And these macrophages usually engulf with the process called phagocytosis. So that way, this was a little easy understanding of how the viruses get into the cell, and how do they multiply, and how do they come out of the cells and get ready with the baby viruses to infect the other cells again. So that way, this is like the clip which I usually wanted everyone to get to know about the exact mechanism, how these viruses get into them. Now a little bit we'll move on to the structural part. Even that has been discussed earlier, but still for a little layman understanding, we have the usually viruses are roundish. There was a concept, like if the viruses are bigger in size, they come up with some shapes, the triangulations, the icosahedral shapes. Otherwise, in the smaller viruses, they have that spherical shape. That is, there is a physics behind it. How do they attain those shapes when they are bigger? Because of the elasticity and the plasticity of the protein molecules which are there on the surface. Sorry. So this was these are the spike proteins, S proteins, or spike proteins they call. This is this is the membrane protein and the proteins in between here, and this is the RNA which is there with the core of that virus. So, and this is an electron micrograph which shows how the coronavirus has. This is the real coronavirus, which has been picturized through the electron microscopy. They have been colored. Now, we have softwares to make them colorful so that we can understand the different portions of those structures that we microphotograph on the T and the electron microscopy. So this is the S protein structure. How does S protein looks like? Even the earlier presentation, it was explained in a very well how molecules are like entangled, how the convoluted molecule structures are there, what it is exactly like. So this is the spike protein. This is the S protein which usually helps the virus to get into the living cell of our throat region, the nasal region, and the lower respiratory tract too. So I think this is the structure. When the molecules, the viruses are a bit bigger in size, maybe more than 50 nanometers. So they attain typical structures of icosahedral with the triangulations, and these triangulations, these apexes, these vertices have usually the spike proteins. Anyways, you don't want to go in detail of those. So that way, it was a bit of structure, and then again we go to a little bit into the process. How these we have seen in the clip. This is a typical X-ray photograph of infected lung. See, the white is not good. More the white, the more is bad the situation. Okay, and in X-ray it comes in a different way. This has been another photograph which has been color micrographs with special techniques. I have already acknowledged in every slide. These yellow-green structures are the coronaviruses, and these yellow-green spots on these epithelial cells, they are the receptors by which they are recognized, and they have been internalized. So we would see this is another three-dimensional X-ray photograph of the lung which has been infected in COVID patient. It was done in some hospital in America. Now, see, so this is what, the internalization, how the virus gets into. This is the S proteins, which have got much of similarity with the receptors. These receptors are there on the epithelial cell of our throat. And this is angiotensin converting enzyme 2. You must have gone through the extensive explanation of how the ACE2 helps out in getting the virus inside. So the layman terminology in layman way. This is the receptor which recognizes the S proteins of these viruses, and these viruses are helped out to get into the living cell for further multiplication, for making copies of those viruses so that they come out and they can infect the other cells too. This is I would tell about this Epoch Times. There's a correspondent from this Epoch Times. He has the correspondent has done an extensive study, an extensive survey on how the coronavirus has emerged from China and how it has spread all over the globe, and how exactly the smaller version of the mechanism also has been depicted in his report. So we would go into the details a little later, but this is how the recognize and the virus has been internalized, and this is typical structures which this correspondent had used in his report. This is how the internalization, the receptors are recognized, and this is how this has been internalized. So anyways, these are the acknowledgements of these photographs and all. Now we would go to a little bit of the controversy thing which has been related to this coronavirus. So first of all, I would like to tell you all, this is a lady where sorry, This is a lady. She is called Shi Zhenli in 2010. 10 years back, 10 years, 9 years back. She was like, she was working on the coronaviruses, not this one, the one which has been spread all over the globe. She was trying. She was like she was one of the persons who had contributed in getting these variants of these viruses. But she was working on the bat coronaviruses, and they were like, they require. Sorry. So. Sorry. Hello.
I request all the participants to please mute their cell phones.
Yeah, thank you.
Thank
so
you.
she Zhengli, she was like working on the bat coronavirus at the time. And she was a very hard worker. She is a researcher by nature, and she was like very much into the research. She did not have any intentions of she has been blamed for some contribution for creating such kind of horrifying virus. But it's not like that. But she was like a dedicated researcher. She was working on bat viruses, and what happened? She wanted to do some recombination procedures on the viruses she was working. So for the recombination experiments and all, they need a specialized lab, which is called P4 lab. So Wuhan Institute of Virology was already there. She was there in the Wuhan Institute of Virology, but she needed a P4 lab. So P4 lab needs bigger fund, much. It's a fat funds they need to maintain those P4 labs. Usually a government used to fund for building those type of labs. Because when you enter in the P4 lab, you do not enter like as we go. Simply appearing a vapor apron and entering into the lab is not allowed. You go as if you are entering in a space shuttle. You're always all covered. You don't breathe inside the lab. You need to have a gas lines to breathe. So you're breathing in, breathing out, two gas lines. So it's a very sophisticated lab where the combinations are done, because you need that environment to be very, very pure, like very clear, very healthy, and they should not have any contamination. Exactly, they should not have any contamination of any other strain or the pathogen or agent or any non-living virus. So they need such kind of. She wanted such kind of lab associated with the Wuhan Institute of Virology. She went many places to for raising the funds for that project to establish a P4 lab. She went to US, she went to UK, she went to the Asian big countries, big institutes, big funding agencies, and she had delivered her ambition, her vision of work, and somehow she got the fund, and it's been the P4 lab has been constructed over there, and she started her recombination work, recombination experiments. And there rose some controversy rose, but I would not like to go into the much. She at younger days when she was working as a researcher in the Wuhan Institute of Virology. We would not go much like controversial aspects of this, but only one should know what Shi Zhengli is the researcher who was working on the bat viruses, and she was trying to recombine some of the COVID viruses, coronaviruses, and she could come up. Even it's been told, it's been under the controversial aspect, like she had made a susceptibility of these viruses to the HIV portions, which are susceptible to human cells. Anyways, we will not go much detail into the controversy. And then, the much much import should be given. It should be given much importance. in terms of, because as we all know, it has come, it has infected people, and much havoc has been created around us, and many people died. But still, this is the latest, I think, the latest figures, I suppose, today's figures. So see, we are reaching around 10 million, right? And 0.4, this is 0.1 million people have died, and almost 8.7 million people have recovered. Only 0.4 million people are still active as COVID patients. So this is the site, this is COVID-19india.org, at which these details are maintained. And this is another this is a very good representation of the data that has been collected. I wanted to tell you this. These figures, this data is just to make yourself aware of the situation, but not to get panic. Because this graph was all was there on this site earlier, on this site, but this has been removed because the people have started sharing panic among themselves, because they thought, because in this graph, it shows how the infection has spread from one person to another person, and from there to another. So how does the chain has been maintained, the chain has been depicted in this graph. So, but later it was removed because it was like a little creating a panic among the public masses. So it was removed. But this is a very good example, a very good representation of the present scenario. But we don't see it. At least we the students of sciences should understand this graph, should know about how these are related, how the situation is there. It shows how the situation is there right now. But we don't. I would also tell, don't fall prey to the false information and the fraudulent videos and information and audios which have been like circulated extensively on this social media. Please avoid. Try to get into the authenticity of that. And another thing is that getting tested. I think we have heard soon the Maharashtra state, like Nagpur state, Nagpur district will be like sealed from its borders, and the people who try to come inside the Nagpur district, they need to have a negative test report, and that too not only the antigen based test, they need to have a RTR based test report. So that this is the RTR based test report, taking the swab and doing the amplification of the genome which is collected. So that way, so this is the type of combating thing. And in previous presentation, it was told the vaccines are being like developed, and the portions and the points on the basis on which the vaccines are developed, that's been discussed very well. So those vaccines have been like the spike proteins are made and they are injected. Only the spike proteins, not the envelope membrane or the RNA. Only the spike proteins are being replicated. They have been synthesized and they have been like injected into you, so that into the persons, in the normal person. These spike proteins are now recognized by our cells, by the receptors, the angiotensin converting enzyme 2 receptors. So now what happens? They have been internalized. But this has been like there is no virus along with it. So our immune system automatically develops as the earlier presentation told, how that B cells are instigated, how that the phagocytes are stimulated. So that way, these things are like they create an automatic immunity, adaptive immunity against these viruses. But what happens? In the clip we have seen, when it is getting multiplied in the nucleus of the epithelial cell, what happens? It happens so fast. It happens so fast. Of course, the fast replication has got a precision. Our copying system is very precise, very accurate. But still, the viruses which come inside and the RNA which is incorporated. Sometimes we have got reverse RNAs which make the DNA, and the DNA is composed, is recombined with our existing one, and then they make copies of it. The different mechanisms of getting copies. So those mechanisms sometimes make these sequences a bit haphazard. They are not aligned properly in some cases, in many cases, or in some cases you can say. So these rearrangements and misarrangements usually cause the viruses with little mutational aspects or changed aspects in the terms of molecules, in the terms of envelope protein molecules, or the S protein molecules. That way, because of some misarrangements, undue arrangements, or the sets, these are like, we find them with different sets of protein molecules arranged on them. So that's why we call that they get mutated every time. So the mutations can be of n number of, it's a multifactorial thing. We can find at n number of places they can get themselves changed in terms of protein molecules they bear, in terms of the RNA which they carry. So, and that's making us more difficult for making a vaccine, because if we have developed some vaccine which has got the S protein resemblance, and after a few infections at some place, if it comes with a different type of S protein which has got the resemblance with the ACE2 on our receptors, so the vaccine would not work. So that in that way, it's getting difficult, because in many places, in many instances, the changed sequences of these certain molecules is coming with these viruses. Anyways, people are struggling everywhere. Researchers are struggling everywhere to develop a good vaccine, more appropriate and more effective vaccine, more potent vaccine. Sorry. So till then, we need to do some precautionary things. We need to do some precautionary things. And I tell you, the viruses, if they are on your hands, sorry, the viruses which are there on your hands, they themselves will not fly off and get into somebody else's. And the viruses, if at all, they are those places where it's in public. In public places, there are many objects which multiple people touch. Suppose staircase rail, right? Or if you go to some the cyber cafe, there you may touch the keyboards, which are touched by many people. There are many things in buses, the steps and the holder by which we hold and we climb up the bus, we board on the bus. So there are many things, the seats, the many public places where we keep on touching the objects which multiple people touch. That time, at least, that time you need to sanitize your hand, because you never know who has come over there, who has touched it. So you need to sanitize it. Keeping sanitization every time, after 10 minutes every time, it's not it is not required. But what is required is when you are touching something which is like in masses, in public, where multiple people touch those objects, then you need to sanitize. Definitely, don't forget to sanitize in public places. And when you're in masses, when you're in the crowded places, the malls, in the bazaars, in the sabji bazaars, hearts, in these places, when you come in very close proximity with these people, when every people, like, not these people, these type, but you never know who is infected and who is not, who is having the symptomatic effect or who is not having the non-symptomatic effect. So you never know that. So it's a precautionary measure. If at all somebody's there in the crowd, you need to protect yourself. And masking is the best protection. Sanitizing, if you're touching something, and if you're not touching anything and you're coming close proximity is considered within the 3 feet region, 3 feet distance. You have 3 feet distance ke andar aate hain, so you are in the close proximity with that person. If that person is infected, you need to protect yourself, because if he talks, he might send some droplets, because some people, while talking also, they spit small droplets. And sometimes by coughing or simple coughing without covering hand over the cough, hand over the mouth, sometimes small sneezes, they all might carry those droplets with viruses if the person is infected, of course. So you need to be precautionary. You need to take care of yourself. And that another thing which usually we do not follow in our country, we because of the crowded nature of our country's masses. It's crowded everywhere, but we need to like socially distance ourselves. But as I always say, why to touch somebody else's biological property? Keep a distance with them. So the social distancing and keeping masking yourself in crowded places and sanitizing your hands, and when you are touching and when you are into the public places where you touch the objects that many people touch, and keeping track of the records, keeping track on the statistics, which do help you in assessing the real situation of the pandemic. Because the thing which is coming up in social media is it's not always right. You need to go for the authenticity of the information that is being circulated. So you need to be a bit aware, a bit learned, a bit educated, a bit non-panic. The first of the non-panic. Anyway, that was all I just wanted to share with you all. Thank you very much for giving me this opportunity to speak before you all and share a few things about the COVID in a different perspective. Thank you very much.
For your remarkable address in understanding genetics and origin of COVID-19 in a better way. So we have one question from the participant.
Okay.
What is the possibility of being reinfected after after affected patient being cured?
Oh, okay. I would say very less chances, but I would not say that there is zero chances. There's one the Apex Institute in US. It's National Institute of Infectious Diseases and something. So the headquarters is in Texas, and the director of that institute, Dr. Alfred Fauci, he said, theoretically speaking, there is no chances of getting reinfected, but there are chances of getting reinfected. Like just like you must have seen, once it's जिन जिनको चिकन पॉक्स एक बार हो चुका है तो कहते हैं कि we would not get chicken pox again in our life. But there are chances. There are cases in which we have learned that chicken pox has again infected in that particular individual. So reinfection is a different case. Once you have already developed an immunity against that particular virus or pathogen, the next time when that pathogen or the virus gets into your body automatically recognizes it and it fights with it. But if your immune system in due course of time gets compromised, that means your food habits, your lifestyle, this all helps in spoiling your immune system. When your immune responses get delayed because of intoxication, regular intoxication also, if you are like subjugated to regular intoxication like alcohol or drugs or smoking, these things usually delay your immune responses. I would not say they spoil, but they delay. So sometimes these delay in responses make you get infected, though you may get cured easily. But reoccurrence is theoretically of no chance. But in humans, you cannot just draw a line that this is yes, this is no. In human cases, there are n number of different species, so n number of chances, n number of probabilities. But the chances is very less.
Thank you, sir.
Thank you. Thank you very much.
The time has come to draw a curtain to our one-day national. Sir, I could request you to please stop sharing your screen.
Yeah, I'm just trying to do that.
Yes, sir.
Why it's getting shared again? Is it stopped?
No, sir. Yes, sir.
Yeah.
So the time has come to draw a curtain to our one-day national seminar on COVID-19 impact of life sciences. So once again, good afternoon to all friends. We have been listening to the eminent speakers deliberatory right from the morning in the different areas of COVID-19 and plant diversity. Hope you all have acquired a valuable insight regarding all the topics discussed. Friends, we all have experienced unprecedented situations with the global COVID-19 pandemic, a virus not visible to the naked eyes, but have put the whole world on pause. It has no boundaries, no race, and no religion. It teaches us that only a global solution underpinned by solidarity and swift action can counter its force. May I now request Dr. Manoj, Head Department of Botany, to propose vote of thanks and also feedback for the seminar. And at the end by saying solidarity is the key to defeat COVID-19. So take care and be safe. With this word, I, Dr. Rupali Tekade, takes a leave from you. Over to you, Dr. Manoj.
Thank you, ma'am. Good afternoon, everyone. All the good things comes to end in life. So is the webinar. So on the behalf of Sri Datta Prasadi Shikshan Prasarak Mandal, Arne, I take this opportunity to propose vote of thanks to those who have directly as well as indirectly contributed to this national webinar on COVID-19 impact on life sciences organized by Department of Botany and Zoology and IQAC of LR Bharati Arts Commerce and SR Bharati Science College, Art. Today, I, Dr. Manoj Kakure, Head Department of Botany, take this opportunity to put my gratitude into words. First of all, I would like to thank Almighty God for making today's national webinar a grand success. I extend my sincere thanks to Honorable Dr. Sanjay Ji Bharti, President of Sri Datta Prasadi Shikshan Prasarak Mandal, Arne, for his presence in the webinar. Thank you, sir, very much for enlightening us with your inspiring and motivating words. At the outside, I am also thankful to our chief guest, Dr. Ravi Kiran Goitkar, sir, Chief Conservator of Forest and Field Director, Finch Tiger Reserve, Nagpur. We are really enlightened with your inaugural remark. Thank you very much, sir. Also, I may like to express my sincere thanks to Honorable Speaker Dr. Mumtaz Bey, Department of Integrative Biology, University of Guelph, Ontario, Canada, for very informative and knowledgeable talk on this event. Thank you, sir. I am also grateful to Honorable Speaker, respected SR Yadav, sir, for enlightening us with your inspiration talk and words. You are an exemplary person and we have a lot learned from you. We all are inspired by your great talk. Thank you very much, sir. Also, I would like to thank Honorable Speaker, Dr. Ashish Kumar Jha, Hislop College, Nagpur, as well as Dr. Devdura Venkat Suresh, Dr. Ambedkar College, Nagpur, for informative and fruitful talk on this event. Thank you very much, sir. Wholeheartedly, I would also thanks to our principal, Dr. R.M. Tater, sir, for his enthusiastic support and guidance for organizing such an event. Thank you very much, sir, for assisting and encouraging all of us. Also, my heartfelt thanks to all the respected professors, research scholars, and student participants from and different states of India and outside of India for making this webinar grand success. A special thanks to organizing committee as well as technical committee of this event. Also, teaching and non-teaching staff of the college for their unflinching support and coordination and enormous cooperation for making this national webinar a grand success. Once again, I want to state that we all are grateful to all the speakers on this event. We thank you for being with us on this day. It has been a great pleasure. Lastly, with the permission of all the dignitaries, I declare here that today's national webinar